words. And I still have some problem in changing slides, but okay, so this is what you all know, the spectrum of our star, the sun, and the part in color is the spectrum of the sun at the sea level. So photosynthetic organisms normally use, most of photosynthetic organisms use the light up to 700 nanometers, and you have seen that this is due to the fact that they have pigments that absorb up to there. Thermodynamically speaking, so there is of course a limit for the wavelength of the photon that can be used for splitting water, but this limit is not 700 nanometer. Only thermodynamically you can use photons of lower energy. At least there have been a lot of discussion for many years, universal potential, this is not possible, far red is not possible, but in principle, far red should be possible. And why is far red actually important? Because light is not having a different color because of different stars. Light can also be of different color in different parts of our planet. And what I'm plotting here is are the spectra of the light at different depth in the sea, in different type of sea, in different type of water. And it's the spectrum when the intensity is 1% of what it was at the surface level. So for example, the blue one here, yeah, not sure it's really okay. The blue one, the first one, is Pacific, in a Pacific Ocean, in a place that is pretty clean. So the light is penetrating very much up to more than 100 meters, and then at 100 meters, with an attenuation to 1%, you have only blue light. When you go to the IJsselmeer, these are a kind of a lake in the middle of the Netherlands that is less clean. You get a green light, but of course the penetration is only 10 meters. And there are a lot of puddles. So this is what I call puddles or little lakes that are very dirty. If you were Dutch again, if you enter in one of these little lakes, you don't see your feet after 10 centimeters of water. So this means that all the visible light cannot penetrate and that you have a far red light only. And I told you photosynthesis occurs everywhere, so it makes sense that there are also organisms on our planet that can use far red light for photosynthesis. And in fact, they are there. And are being discovered. The first type of cyanobacteria can do far red light photosynthesis has been discovered in '96, and has been considered for a long time like an exception. Only is an Acariochloris strain, and the genus Acariochloris is apparently able to absorb far red light in a constitutive way. But the big change, I would say, at least for me, has been later on, first with Min Chen discovering in 2010 a type of chlorophyll that absorbs in a far red, chlorophyll F, and then with the wonderful work of Don Bryant that it was published in 2014, and what it showed that actually you can have normal cyanobacteria that they do photosynthesis, we all know, and then you put those cyanobacteria in far red light, and they change their photosynthetic apparatus, and they become able to use far red light for photosynthesis. For me, this was extremely exciting and a game changer, and then I started also even to work on cyanobacteria, which I didn't do before, and getting very excited about it. So one point is, of course, how can this photosystem work using far red light? Far red light carries less energy, so this means that they are doing the same process, oxygenic photosynthesis, with less energy. And the point is, how to do it and what is required, what are the requirements for this. Before going into detail, I want just to re-stress that there are two important parameters that have to be fulfilled for doing photosynthesis with a certain amount of light. The first, banal, you have to absorb far red light, so you have to find some pigments that absorb in it or some way to absorb it. And the second, that is equally important, is that you don't only have to absorb this light, but you have to also to deliver it to the energy to the reaction center for charge separation. Otherwise, you absorb, you're done. You cannot do anything with this. So we are going through these two requirements. And the first is, of course, we want to absorb far red light. And the first way is to do it chemically. You synthesize a new pigments that are able to absorb far red light. This is what the nature does in this cyanobacteria. There are at least two pigments that are chlorophyll D and chlorophyll F, that they have the lowest energy transition, the important one, at lower energy, so higher wavelengths. And in both of them, there is only one substitution in one of the position in the ring. In one case is a methyl for a formyl, and in the other case is a formyl for a methyl, and the other case is a formyl for a vinyl. And these, of course, are changing, as Jonathan was showing you before, the energy gap between the HOMO and LUMO, and then you have a shift to the red of the absorption. And these are the pigments that are present in this, in this photosystems. What I just want to add about the chlorophyll is that we were really discussing briefly before if chlorophyll have been ever found, or tetrapyrrole have been ever found in this primordial path. But of course, chlorophyll are very difficult to synthesize. You need at least 16 different steps. And you need nitrogen, and so they are complex, they are big. Of course, you want to absorb visible light, you need big molecules, so conjugated double bonds. But what they have that is amazing, in my view, is that with one substitution to the ring, you can really shift the absorption. So this means one enzyme, and you make a completely new pigment, or not completely new pigment, but a pigment that really absorbs a different wavelength. And this, it helps a lot, or it helped a lot the flexibility of photosynthesis, in my view, of seeing it, because then you don't need to reinvent the wheel. You just and maybe this is also why the chlorophyll, they stay. Moreover, they have another great properties, that is the fact that they have a strong absorption, or relatively strong absorption, and relatively long excited state lifetime. The two, there is going together in a different direction normally. Okay, so now the second, so first you can make different pigments. The second is that you are using the protein to tune the property of these pigments. And this is LAC2, is the major antenna protein of plants, and is relatively small protein, 25 kilodaltons, with more than 15 kilodaltons of pigments, 14 chlorophyll, 4 carotenoid, is really packed with pigments. It contains chlorophyll A and B, and you can see there the absorption spectra of chlorophyll A and B in acetone. So of course, chlorophyll B is absorbing at higher energy than chlorophyll A, but all the chlorophyll, the ratio of chlorophyll A and B that you have in the spectrum is the same that you have in the protein. Now, magically, I can take these pigments and put it in the protein, and now you see that the absorption of these pigments is changed. Now it's tuned by the protein, because every pigment is in a different binding pocket, to say it in an easy way. So the pigments are also interacting between each other. They all feel a different environment, and this environment allows to tune everything. Then let's see if it starts. The point is also that these proteins are dynamic. So this is dynamic molecular dynamic simulation. So the environment of each of these pigments is changing in time. And this changing in time is broadening the band, of course, but is also have an effect on which one is the lower energy side. And what you are seeing here is a single molecule measurement. So one is LAC2, and you see that the spectrum is really changing in time, and simply because the environment of the pigment change, and then you can even have shift to the red, because you increase, for example, in some of the conformation, the interaction between pigments. So this is the other way that nature is using for moving the absorption. And I have two examples. The first here is a paper that we published a couple of years ago together with Chris, and this is about bilins that are associated with the antenna of cyanobacteria, the phycobilisomes. And these bilins chemically are absolutely the same, but when they are introduced in different proteins, they assume a different geometrical organization, and then more flat they are, more all the different rings are on a plane, more the localization you have, and then smaller the gap between the HOMO and the LUMO, and more you shift to the red. So the pigment is exactly the same chemically, but the geometry of the pigment changes, and this creates a shift in the absorption. The other example photosystem 1, which has what are called the red forms, are known for a very long time. And these red forms are not due to a change in the organization on the geometry of the pigment, but in a change or in the organization of pigments nearby. So they are getting very close together. They enter in excitonic interaction. So in this way, they are mix in the wave function. And then on top of this, they can also mix with the sun charge transfer state, which lead also to very broad bands that are shifted to the red. So all the theory, if you want it, is explained there. But this allows really to move very much to the far red. From a typical chlorophyll A 660, you easily arrive at 710 nanometers just using this trick. And I just want to show also that the difference between the spectral form, if we look at the effect created by the protein, is small. So from years ago, we discovered that this redshift absorption and emission of photosystem 1 is due to the interaction between these two particular chlorophyll. I'm zooming in now in one of these. And so what we did is just mutate the ligand for one of the chlorophyll from asparagine through glutamine in 1 cc bond more. So fundamentally, we are changing a bit the distance between the two chlorophylls, but far less than 1 angstrom. And then the spectrum is becoming blue. So sometimes the structure don't even have a resolution good enough for seeing this variation. The spectroscopy is very sensitive to this, and very small variation can have a very large effect on the absorption and emission of the pigments. And so this is an example for, and you go back to our molecular dynamic simulation and you see a change in the organization of this pigment. This can easily explain because you go from a J to a H aggregate. Okay, what is the most red absorption that has been seen up to now in an organism doing oxygenic photosynthesis? And to my knowledge is this little band there. That is almost 800 nanometer. The maximum is 790 nanometer. Is a chlorophyll F inserted in the photosystem 1 of this far lip cyanobacteria that Don discovered. And this, the origin of this really red shift is because there is a chlorophyll F that is far red chlorophyll, and because the protein environment is influencing it. And then we also could pinpoint in the structure of CRIS which one was this. These are the chlorophyll B37, B38. Okay, now we have absorption is done. We can absorb a far red light up to, let's say, 800 nanometers. And now the second problem is to deliver it to the reaction center, because, of course, otherwise it's all lost. So this is a representation, is the structure of photosystem 1. You see there are many proteins, different colors, and many pigments. There are 100 pigments in this photosystem 1. And now this is my movie is a bit slow, sorry. It will just now show the organization of the pigment from the top of the membrane, and all this green are the antenna chlorophyll, and the red there in the middle, or the bulk in the middle, are the reaction center where the energy has to go. And so what is fundamentally, happening is that the energy is absorbed by, the photon is absorbed by one of these 100 chlorophyll, only one, because light is a diluted form of energy. You never have two excitations, even on our planet. And then it has to be transferred to the reaction center, where it leads to charge separation. Now, how fast it's transferred to the reaction center for the charge separation determines how efficient the system is. You have in the first equation on top there. So how fast depends on two parameters, on two processes. One is how long it takes from the energy, from when it's absorbed somewhere to when it's delivered to the reaction center. This is migration time. And then the second is the trapping time, that it means it depends on how fast the energy is trapped. So the charge separation occurs when the energy is on the reaction center. And yeah, you can see the equation that is multiplied, this tau CS is multiplied by N, and N is the number of isoenergetic pigments, because it's also the probability, it contains the probability to have the energy on the reaction center. In other words, the larger the antenna, the longer the time. And how in a is this influence in the efficiency. The equation on top is 1 minus this T average, that is time that it takes from when it's absorbed to when it leads to charge separation in the reaction center, divided tau chlorophyll. And this is the time that Jonathan before told you is 6 nanoseconds, is the 1 over the other rate of decay of our chlorophyll. So you fundamentally, what you do, you have to deliver the energy to the reaction center fast enough to beat all the other decay processes that are all occurring, but they are far slower, so the rate is hopefully far smaller. And this defines your efficiency. Now let's look at the photosystem 1 and photosystem 2 from the different cyanobacteria that I was mentioning before. So the one in black will always be a normal cyanobacterium, the one with chlorophyll A only. The one in red is the one containing chlorophyll F, so it's a far lip cyanobacterium. The far lip photosystem from a cyanobacterium. And the chlorophyll D one is an Acariochlorus, are the system from Acariochlorus marina that they contain chlorophyll D. The first thing that you can see is these are photosystem 1 on the left and photosystem 2 on the right. The first thing that you can see that for also the chlorophyll F containing cyanobacteria, most of the chlorophyll, actually chlorophyll A, is the absorption is the same that our chlorophyll A, PS1, and there is only a tail with few chlorophyll F that absorb in a far red. And interestingly enough, this is enough to make the cyanobacteria to be able to use far red light. Of course, they don't grow like rockets, but they grow, they do photosynthesis. So the other one, chlorophyll D instead, is all the absorption is already far red, but is less far red than in the far lips. And this is identical for both photosystem 1 and photosystem 2. Now, how can we calculate or can we see if they are efficient or not? We can do time results fluorescence, so we do time results spectroscopy in my lab, combined with molecular biology. And then we can excite this system that can be isolated complex of the cyanobacteria, whatever. And then we shine light on it and we collect the fluorescence. Fluorescence is a good reporter for whatever else is occurring because it's one of the decay pathways. And then we have a picosecond resolution, so we really can see what is happening in the system. You have this map in color. What you have is wavelength by time. And then if you cut like this, you see the decay of the fluorescence, and you cut like this, you see a fluorescence spectrum in time. You analyze all of this, and you can extract the time that the energy is transferred from one part of the complex to another, and also how long it takes for the decay. I have 5 minutes. I will then just go very quickly because so what we have here is that from this time that you can calculate the trapping time, let's say how long it takes to be trapped in the reaction center. You can see that in a normal photosystem 1 is a probability 98%. Out of 100 photons absorbed by the system, 98 lead to charge separation in the reaction center. It's the best energy conversion in a system in nature. If you go with the chlorophyll F containing one, then it's lower, but still, what is 92% is trapped, not too bad. And with the Acariochlorus marina, everything is lower in energy, but then you have still super high, 98%. What I wanted to show you very briefly, and then now I have a bit of a problem with the time, I have to say, is that we spent a bit of time before, so because I want to give you this concept that is important. In many papers that you're reading is written that energy is transferred only from high energy state to low energy state. It is not true. You can also transfer in from a low energy state, so from this red phone to the reaction center, if the temperature is high enough to provide enough to cover this gap, or even more. You have a protein also helping to push up these transitions. So what you have here, for example, is in this system of far-rip cyanobacteria. We found out that the reaction center is around 750 nanometer, absorb around 750 nanometer. And you remember we have these red forms at 790 nanometers. It's still efficient in transferring energy. Actually, it's extremely efficient in transferring energy to the reaction center at room temperature because it's located very close. These chlorophyll are located close to the reaction center, and because there is this thermal energy that is helping. If you go to low temperature, 77 K, there is no way that you transfer this energy uphill. So the temperature of your planet it will also influence what is possible to do with this far red light. Quickly to photosystem 2. The story in photosystem 2 is a bit more complicated. The photosystem 2 is very difficult design because it's doing this super chemistry to take an electron from water. It's super oxidizing. So as the organization of the big is slightly different. Photosystem 1 is really rock solid complex. Photosystem 2, if we do the time resolve, we observe that the time it takes for the energy to be trapped in the reaction center when you have chlorophyll F around is far longer than in a chlorophyll A containing system. And the efficiency indeed goes down to 65–70% when instead it was around 92% in the real system. So what is the reason why using far red light photosystem 2 works less well or is less efficient? And there are two options. The first two possibilities, both of them are actually occurring here, one is that there is more a slower and more reversible charge separation. I'll show you in a moment. And the other is that it's taking longer for the migration to reach the reaction center. So first, charge separation. What is happening here? This is the scheme that you have also, I think, seen before, and they also have the nice poster of Govindjian outside. So what you have is the photosystem 2 is the reaction center is excited, is P680 for a normal photosystem 2, and then there is charge separation, and then the electron is transferred to a primary donor, secondary donor, and so on. So what this photosystem they do, they have a charge separation, and then they take this charge as far as possible, as fast as possible, and then every different step they also lose free energy, Gibbs energy, to avoid the recombination, because otherwise the most probable thing that you do is just to recombine and you have lost all the energy. And of course, when you have a reaction center that is far red, then you go from 680 to 727 nanometers. So this means that you are trying to do the same chemistry with less energy. Everything is squeezed. And we know, because water splitting works very well, that at the donor side, there are no problems. So the squeezing is occurring at the acceptor side. And there is a price to pay that is more recombination, or some people even say more photo damage. This is something that we are still analyzing. But anyway, you pay a price. You are trying to do something with less energy, of course. The second point is the migration. What is happening here is also representation of photosystem 2, and on this side here, you see the energy landscape. It's very flat. So what it means that most of the chlorophyll are very isoenergetics with the reaction center. Energy is flowing to the reaction center in around 50 picoseconds. It's fast and very efficient. Here also you see the profile. If you have a chlorophyll F, I showed you that you have only a few chlorophylls F embedded in many chlorophylls A. And now your profile is very different. And now it's not anymore isoenergetic. You have this very low energy chlorophyll in between that are then acting as a trap. Energy is going into this trap and it's coming out again, because at room temperature we have thermal energy. But if you all know Boltzmann, so the rate on energy transfer from high energy to lower energy is far larger than the uphill. And this means that everything is becoming slower because it takes more time to get out of this little hole. This is also working very well with the structure of Chris. I have no time to really go into this and to explain how the system as I finished. What they found, but I only want to show you our next calculation. So what we are trying to do here is to understand how many chlorophyll at low energy we can put in a photosystem and where we have to put it, because this is the other important thing. I didn't have time to explain you this. So we want to know where and how many and where. And this is a difference that you don't know if you can read these are a number of substitution of chlorophyll A per chlorophyll D in the core of cyanobacteria, and these are the lifetime. So you really change the lifetime extremely much depending on the number of chlorophylls that you put in at low energy, but also depending on where you put it. And this one are 3 or 4 different trials in which we try to put a low energy chlorophyll in the reaction center in one position, 2 position, and 3 position. And you can see very well that if we keep our 680 chlorophyll in the reaction center, immediately our lifetimes is going very high to 1 nanosecond. So we lose already half of the energy that we have absorbed because we don't have a way to transfer it in an efficient way. So with this, I have a few take-home messages. In principle, far-red photosynthesis is possible, but there is always a little price in efficiency. How big this price is depends how the system is designed. So the number and the position of this low-energy chlorophyll is extremely important. You can even get a system that is really not working at all. You absorb the light and is completely gone. The reaction center limit, so what is the limit of oxygenic photosynthesis? I can tell you that the reaction center are only seen for photosystem 1 and 2 up to 750 or nanometers. So there is nothing above 750 nanometer that we know now in the reaction center. But the antenna is much more flexible. We can go even up to 800 nanometer, and who knows, even more. And in my view of seeing this is the limit of oxygenic photosynthesis. It means if I can absorb, or if they can absorb 800 nanometer and use this energy efficiently for charge separation, then I see the limit there. Other people have a different definition of limit that is the one of the reaction center, but I don't mind if we needed to use some energy in the environment to push in this transition. I think can still do photosynthesis. And then, there are many factors that are influencing the efficiency of the system, and this needs to be taken into account in the design. And the temperature of your planet also then plays a role there. And with this, thank you very much, and sorry for all the technical problems before.
Thank you for that excellent talk. We have time maybe for 1 or 2 quick questions as we load the next slide. Any questions in the room? Nancy, we have, but if you can load that next one. Okay.
Junkoyama from Lawrenceburg. Thank you for beautiful talk. So I understand that dynamics plays a very important role. So you do probably have a temperature, variable temperature dependent data. Is that like a broadening, continuous broadening, or jumping from one absorption energy to?
No, if you what you see here is the effect on a complex in which you have many different chlorophylls. So the band are getting narrower with the temperature, but then what is happening is that the conformation is changing, and then you change the absorption of the pigments. Because maybe then you create more exotonic interaction, and then on a different part of the protein.
Any other?
Thank you for the nice talk. Very quickly, so you mentioned that the time that it takes to transfer the energy is actually kind of proportional, inversely proportional to the efficiency. That I suppose is quantum efficiency.
It's always quantum efficiency in photosynthesis.
So I was wondering that this time, the kinetics usually is not directly connected with efficiency. So I suppose that during this time there are other processes which are competing,
Yeah.
and therefore those
She
are
had
the
a-
ones that are basically decreasing. So is that internal conversion or something?
They are all what Jonathan was saying before, internal conversion, intersystem crossing,
Also
and
intersystem
fluorescence.
crossing.
Intersystem
And
crossing,
emission as well.
yeah, and
Okay.
intersystem crossing is the one with the highest rate between the three of them. It's what this Jonathan would say that is dangerous, and they are always happening. It's
That's a
The
slide.
intersystem-
She was having a terrible time dancing.
the one with the highest
I
rate.
thought it was internal conversion. It was usually.
No, the internal conversion for the chlorophyll is the smallest of all.
Okay, because it's rigid.
Yeah.
So we can continue
Doesn't it?
conversation at the break.
There we go, okay.
So next up, and we'll do a quick mic switch. Next up, we have Barry Bruce talking about photosystem 1 at the edge when photosystem 1 breaks the rules. So this is a 10-minute talk, and I'll give you a quick 2-minute one.
Okay, thank you. I will hopefully be able to stay within the time. Thank you for inviting me, and I thank Roberta for giving such a nice introduction. I'm going to talk about an unusual form of photosystem 1 that you see here that actually was somewhat serendipitous. I was working in an area of applied photosynthesis, and I wanted a more extreme thermotolerant organism, and I looked into the literature, and there was a group of people in northern Thailand that was working with a very unusual species called Crocosidiopsis TS821. And this was an organism that had really robust growth properties. It came out of an unusual type of hot springs in very northern Thailand. And it is in the family of cyanobacteria that Roger Stanier called the Plurocapsales. And if you see right there in the green is sort of the morphological constraints of what makes a Crocosidiopsis. And very interesting, it's one of the few organisms that can undergo binary fission in all three planes. So it's a very unusual, can make a large, very like a Rubik's Cube structure. And this organism did not exist in any culture. So I literally went to Thailand and I met with the scientist that was able to isolate this, shown there, and we were able to bring this organism back to my lab. We started characterizing it. It truly was a very extremophilic organism. We could expose it to very high temperatures, up to 60 degrees. It would live very nicely. We could put it at 4 degrees. If you do this with Thermosynechococcus, it will die within a half hour. You can give it full salinity of the ocean. You can bubble CO2 up to 20%. You can dry it all the way out. This is a phytoagar plate that was dried out and then rehydrated and it comes back. So this is not surprising. The Crocosidiopsis group has been pioneered by many of the people at NASA. If you go all the way back to Lynn Rothschilds and all the way back to Daniela Billy, this group has been a kind of a hallmark of highly extremophilic phototrophs. I didn't know much about that, but when we got that, we decided to try to figure out where it was phylogenetically. And it turns out that the genus Crocosidiopsis is probably polyphyletic. And you can see this is a nice image that you can see based on ultrastructural morphologies of all the different clades. It fits in that clade 2. And as you can see, it's very unrelated morphologically to the heterocyst and filamentous cyanobacteria. I show you in this little bracket, which I call the heterocyst and close relatives, that at least 2 different clades or 2 different groups of Crocosidiopsis can be phylogenetically placed in with the much more classically well-behaved branched and non-branching heterocyst-forming cyanobacteria. If you go to this scientist, Mr. Boudel, he's sort of the father of the phylogenetics of this, and you can see that again, the Plurocapsales breaks out quite differently. There's 2 clear clades of Crocosidiopsis, and then the blue is where my organism sits. So it does not even fit with the most breadth of that. When we started working with this, everyone in the world thought that cyanobacterials were exclusively trimeric. This is an adaptation based on the work that was done mostly in the Thermosynechococcus elongatus. But when we tried to isolate photosystem 1 from that, it was extremely difficult. And eventually we concluded that it is not difficult because it is not a trimer, okay? And you can see that if you range the detergents, and we worked with dodecyl maltoside, but we had to actually test 13 different detergents. PS1 at no level can be isolated as a trimer. It's always isolated as either a tetramer, and if you pull the tetramer apart with high detergents, it becomes a very stable dimer. But the dimer really doesn't break down to a monomer. So it's either a tetramer or a dimer, and so it turns out to be an equilibrium of dimers of dimers. So I then wanted to sort of explore how wide this was. I went back to Paris, same place that Roger Stainier was, Rosie Ripka, who was his longtime curator, and here, Muriel Gurger. So we were able to dig into their entire repertoire, and mostly with Rosie's help, we were able to pull out 60 different strains of cyanobacteria. I took 60 strains back to my lab, I grew them, I put my poor graduate student onto the task of isolating thylakoids and doing exactly the same thing to decide which of these cyanobacteria were trimeric, which were tetrameric. And this is just a small snapshot. Occasionally we would do the microscopy to confirm it, but everything on the top you can see is tetrameric. It goes above that dotted line. And on the trimers, it goes always below it. So trimers and tetramers are distinct, and there's a lot of cyanobacteria that are tetrameric. So the fact that we were working with the Pasteur Institute meant that we had the genomes of all of these organisms. So we could go in and then, I didn't cover this, but in the top, it turns out that the genomic organization of the photosystems are differentially assembled in a different array in the trimeric forms and the tetrameric forms. So that's the different blue balls. But through characterizing all of these biochemically, we could identify that, in fact, not just phylogenetically, functionally, all of the photosystems in the heterocyst and close relatives really were the tetrameric form. So this is a very widespread group of cyanobacteria. So the question is, what derives that tetrameric form? So we did many studies, but the only study that showed any meaningful influence is light. So as you can see, if you grow it at low light, you get mostly a dimer. If you grow it at high light, you get a tetramer. You get a great increase in carotenoid pigments, which I won't talk about for time, but turns out there's unique carotenoid binding proteins that are unique only to this class of cyanobacteria, unique carotenoids as well. So we were able to try to isolate this complex. We did it first with odecyl maltoside and did a structure, but my lab has been pioneering non-detergent isolations of membrane proteins, a class of proteins called styrene maleic acid copolymers. And we could isolate this tetramer in a non-denatured or non-detergent form. You can see here, traps a very large number of detergents. I'm sorry, very large number of lipid molecules, probably between 800 and 1,000. There's quite a bit of density right in the center, which we've not been able to resolve. But the interesting thing was when we did the cryo-electron microscopy, and we could actually get classifications of particles that were in two different conformations. And as I said, this was a dimer of dimers, but at one of the interfaces, this thing was breathing. It was folding like a little set of wings, okay? Very nice. It gives you about a 12 to 13 degree bend. And if I went back and looked at the most closely related atomic force microscopy, which sticks these membranes from a different Crocosidiopsis on an atomically flat mica, you can get all the spacing of these. So by using that same spacing, we put this thing together in the structure that we see, and very clearly you get a nice invagination or you get a curvature. So it looks as if these proteins, and you can see how dense they are in the AFM, if you put these things close enough together and each of them is curved at this 12 degrees, you can build something that's very curved. So then we went back and we looked at the thylakoids of the cyanobacteria grown at low light to high light. And sure enough, if you grow them in low light, they're very flat, they're very planar. They look like everyone's cyanobacteria. But as you go up to high light, you start getting these circular ensembles of thylakoids. So very interesting. 1 minute. Okay, so that's all I'm going to say. I want to take time to particularly thank Bob Blankenship. I would have never predicted 50 years ago that he and I would be sitting in Wisconsin talking about the evolution of photosynthesis, but Bob was my first graduate mentor, and this was actually my second publication, but my first with Bob. And so I want to thank him for stimulating my interest and allowing me to continue for so long. Thank you.
Thank you. You were perfectly on time. Do we have any questions?
I have a question. Go ahead. Go ahead.
Very nice process, Kato-san. I want one question. I know this is more related to the house, almost like we talk about harvesting. Do you think if you use the highlighted and low-light determine the formation different oligomer of the complex? How about the mutants without phagocytosine? Do you think they form more like a tetramer or something like that?
I can say we did one thing. We put the tetrameric form of PSA-L into Synecocystis 6803, and it made a monomer, okay? But you gotta realize we're going from a thermophile to a mesophile. The lipids are completely different. They're highly saturated lipids in the thermophile. They're unsaturated in the mesophile. There's some other small changes in PSA-I and M, but PSA-L is considered by almost everyone to be the responsible component that leads to the trimerization. And in our case, it's also responsible for the dimerization. Okay.
Yes, and one other quick question.
Do you have a question? Yes.
I just want to make a comment. You see the same thing happening in plant leaves, but with the morphology flatter versus bigger for low light.
I'll just say one thing since I had it on my last slide and I didn't get a chance to talk. If you look at the glycophytes, which are thought to be the most basal lineage for photosynthetic eukaryotes, they too have a tetrameric PS1. So very interesting that Cyanophora is tetrameric, although the geometry of that tetramer is somewhat different than ours.
Great, thank you. All right, next up we have Chris Gisriel, and he is going to be talking about diversity and limits of photosynthesis on Earth. This is a 15-minute talk. I know you're going to be perfectly on time, and I'll give you a warning at 3 minutes.
I don't know where that came from. It's a new one, right?
Why is it? Okay. Here.
Hey everybody, while that's being fixed, I'm Chris Gisriel. I'm an assistant professor at the University of Wisconsin-Madison. And yeah, so I'm typically what I like to imagine as a hardcore photosynthesis researcher. But today, really, I made this talk thinking about the astrobiologists that are present in the room and perhaps the astronomers. So it's going to be kind of simple on the photosynthesis side, more of a bird's eye view of some of the ideas about photosynthesis and especially the evolution of photosynthesis that I want to describe to you. So first of all, as has been described in detail, I like to show this slide, just giving you the idea of what it means to have pigment protein complexes that are going to harvest light. So you have all of these pigments, you have some kind of excitation of a pigment, and then there's energy transfer. This general view looks different in different organisms in terms of protein composition, especially in the antenna. But again, the idea is light absorption, energy transfer, and eventually electron transfer. And electron transfer is the moment of converting the energy of light into chemical potential energy to drive metabolism, right? And that sort of occurs where I've placed these red boxes here in what we call the reaction center. Now, I want to clarify a little bit of nomenclature for you. Some people will refer to the reaction center, like Roberta was doing, as this sort of central region in the photosystem, the part that does the electron transfer processes. But then very often in anoxygenic photosynthesis, people will refer to this entire protein complex as the reaction center. So I'll do my best to make those distinctions. But here I'm just showing you different types of reaction centers, okay? I've highlighted on the left the ones we very commonly talk about that are involved in oxygenic photosynthesis, photosystem 1 and photosystem 2. Photosystem 2 is the only one that we know of that actually does the water splitting reaction to produce the oxygen that you and I are breathing. But there's lots of other reaction centers, and they're not all shown here, but they're involved in anoxygenic photosynthesis, okay? And while these look kind of different, if you look at their molecular structures, which I personally like to do because I'm a structural biologist, you can look at kind of the center of them and say, oh, actually, they're all very similar. So this is a figure, this is, I think, my favorite figure that I've ever made. This is when I was a graduate student with Kevin Redding. But essentially what you're doing, you're looking into the center of those complexes at the reaction center pigments, or I should say cofactors, And what you see is that in every one of these cases, you sort of have 5 transmembrane helices from one side, 5 from the other, and they enclose these electron transfer cofactors. So what's very clear about this is no matter which one of these reaction centers you're talking about, they all are derived from a common ancestor. Okay, and you can start thinking a lot about the evolution of these proteins because of these sort of pictures. Because of that, people will often look at these core polypeptides that make those 5 transmembrane helices that enclose the reaction center, and think about how they relate to one another to infer evolutionary relationships. And you see a number of pictures like this in the literature. Bob certainly has some in his publications. Kevin and I have some. This one is from Tanay Cardona that I think is a very nice kind of simple view. Where's this pointer here? And what you can see is that these are sort of the extant reaction centers, if you will. Again, this is kind of based on those core polypeptides. Some of them are heterodimeric cores, some of them are homodimeric cores, but you can think about these and say, well, what must the ancestors have looked like? A heterodimeric core would have been a result of gene duplication and divergence, right? So we can think about what the characteristics of this ancestor to all modern reaction centers would have looked like. And so I won't make the distinction between types 1 and type 2, but it's a thing that you can do. And the only reason that I say that is because you can start taking those core polypeptides, and this is just type 1 reaction centers, but when you actually look at their phylogenetic relationships, you can say, well, okay, in oxygenic photosynthesis, you look at one of the core polypeptides and the other core polypeptide, they're all pretty much similar. Whether you're talking about cyanobacteria or this tree right here, like they're all very similar to one another. You move away from the reaction center, that starts to not be the case so much, but they're all relatively similar, at least in comparison to these. These are the ones that are in anoxygenic photosynthesis, and they are far, far more diverse in their molecular makeup, in the environmental niches where you the organisms that host them. So I think that's kind of a really important point, is there's sort of a lot of flexibility in anoxygenic photosynthesis that you don't see quite as much in oxygenic photosynthesis. I've kind of baked in some questions that I have to the astrobiologists, right? So first of all, is anoxygenic photosynthesis, is it more flexible to different environments? I think there's a very good argument for that. But then you might say, well, when we're looking for life on another planet, and that planet sort of deviates, or at least the resources on it deviate from what we see on Earth, is anoxygenic or oxygenic photosynthesis more likely? To me, the answer is probably anoxygenic photosynthesis. At least that's what we see on Earth. Of course, that doesn't address the issue of detectability, but it's just a consideration. All right. Okay. Ooh. Really? Out of nowhere. Out of nowhere. Try it. Just try it here. Oh, okay. I'll use those arrows from now on. Weird. So the other thing that's important to think about, did that actually do the right thing? Yeah, the other thing that's important to think about is like, what is oxygenic photosynthesis? And different people may have different answers that are probably dependent upon the scale in which you're talking about this. But to me, do you have photosystem 2 or not? You know, are you making oxygen or not? So that's what oxygenic photosynthesis is in some sort of small scale. Here's photosystem 2. Roberta kind of showed a similar picture a little bit ago. And she said, okay, well, this is a dimeric configuration. Each of these subunits, I think she said 40 subunits. So in the monomer, there's 20 subunits. It's a really large protein complex, more complex than even photosystem 1, but certainly way more complex than all of the reaction centers that we see in anoxygenic photosynthesis. So there's some interesting complexity there. At the center where I've placed these stars, you can see this metallocofactor that's involved in the water splitting reaction. And what you have in photosystem 2 are these different water channels that allow water to go into this active site where this metallocofactor is. And at that metallocofactor, you take water and you're essentially using that as your substrate to do this multi-step catalytic cycle where you require light. There's electron-driven proton transfer, and it's a very complex system. That's the only thing I wanted to sort of express about this more than any other reaction centers that we see. Another challenge with this is that you produce oxygen. This is potentially damaging to other proteins, and because of that, not only is that photosystem in itself complex, but you also have an elaborate assembly and repair mechanism for photosystem 2. This is from a nice paper from Mark Novacek's group some years ago, but I have to say this is probably a very small fraction of what is truly going on with this assembly and repair process for photosystem 2. All right, so what is actually needed to drive water splitting at photosystem 2? Heterodimerization, we know that. We can see from the structure that you require a heterodimeric configuration in order to bind that cluster. Probably 2 photosystems that run in tandem, or at least that's what we see here on Earth. A high redox potential, so we're talking about 0.8 volts, that's substantial. About 750 nanometer light, I think Roberta mentioned that too. I agree with that arrangement. And then multi-electron chemistry at a complex metallocofactor and pretty complex repair mechanisms. It's a big ask, right? It's a tall order, I would say. But because you have this abundance of reducing equivalents from water, once you do have that, assuming you have enough water around and assuming you have high enough energy light, then obviously oxygenic photosynthesis would really dominate all of the other types of photosynthesis, which again is exactly what we see on Earth, right? However, I still can't help but think to myself, is it still very unlikely to be able to actually have all of those requirements, that demand to be able to form that process? If you look back at this tree that I showed you before, so these are the 2 that are involved in oxygenic photosynthesis, and that means that all of these photosystems back to the ancestral one would not have been involved in oxygenic photosynthesis at all, right? And none of them require, whether we're talking about extant or past ones, none of them require any of those considerations that are really demanding. So again, is oxygenic photosynthesis unlikely compared to anoxygenic? And I know this is a little bit, I don't want to say controversial, right? People build their careers off of looking for oxygenic photosynthesis on other planets, and I'm kind of, probably making those people unhappy. But I think an important point of this conference is to address some of those tough questions. I've been thinking about this stuff for like 6 months, right? There's other people that have been thinking about it for a lot longer time than me. So please, I very much encourage, discussion about this sort of thing, especially in this, this is not public, but in our group, because I think throughout the talks that have happened so far, we've gotten a little bit of that, but maybe not as direct as I'm putting it here. Another thing I want to mention is what light is actually required. So Dan was showing his bacterial chlorophyll B structure. So this is whole cell spectroscopy. We're looking at those organisms absorbing light out beyond 1100 nanometers, whereas this is the most redshifted example of oxygenic photosynthesis where you're only going out to, let's call it 800 nanometers for absorption to drive oxygenic photosynthesis. I don't know enough about the light availability from other stars to be able to say exactly what the consequence of this would be, but it feels from what I've heard like bacterial chlorophyll-based photosynthesis would be more likely. But that kind of opens up a whole new issue. One of the things that's been an open question in photosynthesis research for a long time is thinking about whether this ancestor used chlorophyll or bacterial chlorophyll. Now, all of anoxygenic photosynthesis today uses bacterial chlorophylls. So by looking at a simple scheme like this, you would probably say, well, probably this ancestor must use bacterial chlorophyll, right? But then there's a problem with that if you're somebody who studies bacterial chlorophyll biosynthesis. So if you look at this sort of the last steps of chlorophyll biosynthesis, you see that in anoxygenic photosynthesis, you use this enzyme to reduce ring D of this tetrapearl system called protochlorophyllide. And then this is basically your chlorophyll, okay? Well, it's the exact same thing in anoxygenic photosynthesis, except for that there's one more step. This lichor reduces ring B that was mentioned a little bit earlier. So because there's one extra step, you might say, well, okay, so that must mean that this simpler one is actually ancestral. And there's a thing that people refer to called the Granick hypothesis. It says stepwise evolution of progressively more complex pathways. I can barely see my slide there, but you get the idea, right? You know, you would have to make some additional enzyme in order to actually follow the progression toward making this bacterial chlorophyll. So that's why it's been this kind of conundrum, right? These two observations sort of contradict one another. And one of the things that we're doing now is we're looking at the actual enzymes, and this actually arose from a number of conversations that Bob and I had. We're looking at the enzymes that are responsible for these steps. In a simple way of looking at it, one of these enzymes is reducing ring D, and the other one is reducing ring B. And when you actually look at their compositions, you start saying, oh, they're pretty similar, and sure enough, those enzymes are related. And you would look at this and you would say, well, probably the one that appears more, let's call it ancestral, would be this lichor. But when you actually take the sequences and do some phylogenetics, it's very clear that this lichor that is involved in the last step of bacterial chlorophyll biosynthesis comes out to be more ancestral compared when you look at both of those proteins. So that's really interesting, and you have to say, well, is it possible that somehow things have evolved in a way in which long ago it was that ring B that would have been reduced first by an ancestor? So working with Betul Kachar's group at UW-Madison, we've actually calculated the ancestral sequences, and we're trying to what they call resurrect some of these ancestors, right? We're trying to make it so that Synechocystis 6803 is actually making these proteins. Okay. So with that, I'll stop talking. I just wanted to present a little bit of our research. I'll thank my whole research group. I'll thank you all for allowing me into your wonderful community. And I'm sorry I took so much time.
That was a fabulous talk, translating hardcore photosynthesis research for planetary scientists and astronomers, asking some provocative questions. We have time for one quick question. See over here.
Greg Fournier, MIT. So I didn't realize those biosynthesis proteins were homologous. That's really cool.
They're also homologous to nitrogenase.
Is that what you use for the outgroup to determine? Okay, because I was wondering, from the figure you showed, I didn't see how you could decide which was older and which was younger.
Yeah, because I had 30 seconds to say it.
But that is
So if you calculate their ancestral sequences, you can simply look at the sequence identities between the extant sequences compared to the ancestral ones, and you always get higher sequence identity with the lichors, the ones that reduce ring B compared to the ancestors.
I see. So it's based on evolutionary distance.
Yes.
Thanks.
Good.
Thank you. And next up, we have Hazel Barton, who's going to be talking about using caves to test the energetic limits of oxygenic photosynthesis. So go ahead and take it away.
Thank you. I appreciate the invitation to come here. I'm quite at my depth. It does look like I'm a geologist, but I've not actually had any formal training in geology. I'm just a geomicrobiologist now, so I work in a geology department. And I did actually think I had 15 minutes, so got quite a few slides. Brace yourself, because this is going to be fast. I want to talk about the conditions that you expect to find in a cave, and these are the conditions that you are normally under, right? They're completely aphotic systems. The kind of caves that we work in are technically complex, very deep. We're trying to understand some of the geochemistries that form that allow these caves to form. Sometimes we're camping underground. We could be underground for many days, and often we're using a lot of technical rope work. It is absolutely dark and it's absolutely aphotic in there. I was given a presentation in Copenhagen about the work that we were doing in caves all the way back, I think in 2015. And Lars Brenhent, I have a hard time with Lars' last name, Brenhent, came up to me and he said, have you considered photosynthesis in caves? So I did the very much like, caves are dark, there's no light in there. And he said, why don't we go look? And so this is Lars with his biofilm that he scraped from the inside of the cave, feeling very full of himself. This is what we normally think about with photosynthesis in caves. So this is something called lantern flora, and this is caused by contamination of the inside of the cave by light. You'll see this where they have commercial caves and they just have regular lights. They don't do anything about the incandescent light bulbs to limit the photosynthetic wavelengths. And so you get stuff growing. This cave is actually proud of their lantern flora and consider it featured. It's incredibly bad for the cave because it erodes the surface. This is the only thing I think about of photosynthesis in caves. But Lars had actually isolated the chlorophyll F organism from a near entrance in a cave. And I believe this was down in Australia. And he said, let's go look to see how far we can go in and what we see. So this is from Carlsbad Caverns. This is the tourist trail. You go around a corner. It's a very dark spot, and you can see just how green it was here. And what we believe is happening, as you can see the entrance behind, is that light's coming off, is hitting the far wall, and then getting bounced into this alcove. And the biofilms can get pretty dramatic in there. Some of those photosynthetic biofilms are up to 2 centimeters thick. So a lot of microbial activity happening. Now that was right near the entrance, and we kept going deeper into the cave. So this is kind of a false image because it's a long exposure, so you could see light. They're lit literally by the light reflecting off the wall to highlight them. And we went deeper and deeper into the caves, and we're still seeing photosynthetic pigments. So even a long way in where you need a light to be able, a headlight to be able to see around, there was still photosynthesis happening. And it got a lot darker than this. This is actually in a cave, and we are 2 corners around. So it's completely dark. You can't see your hand in front of your face, and you've actually gone around true turns, so you cannot see the entrance. This is probably bacterial chlorophylls, but you can see if you were to imagine pointing towards the entrance, you could see that this formation here is creating a shadow, and this bit is being hit by light from the entrance, even though it's perfectly dark in there. Measuring the amount of energy in these systems, you can see you have, this is the photosynthetic active radiation at the entrance. You can see you can have natural daylight here. As you get into the twilight zone, this is where it's difficult to move around without headlight. You're dramatically reduced, and then into the deep zone. So here we're getting 0.03 micromoles of photons per meter squared, and we're still seeing green pigment. The big thing is if you look at the type of light that is there. So this is looking at the near infrared, and you can see you still have a relatively high amount of near infrared light there, even though the amount of PAR light is greatly reduced. In some of these locations, we were seeing up to 625-fold enrichment of near infrared light over visible light. So there's a dramatic increase in the near infrared. Trying to understand what was happening there, I took a rock and we looked at the reflectance of the limestone itself. And it turns out that right at about 700 nanometers, calcium carbonate rock that the cave is made of, this is the rock, so it's a little bit noisier, and this is the pure crystal. It actually starts to reflect everything above about 700 nanometers. So you're reflecting into the near infrared. So just to kind of hammer this home, this is an image I created with a chat GPT. caves are a hall of mirrors to near infrared. So the near infrared is actually getting propagated deeper and deeper into the cave, and you're actually getting fractionation. So you're propagating the longer wavelengths further. So a lot of times when I talk about the science and the microorganisms that we study and what we're trying to understand, I talk about dark life. But when I think about photosynthesis, what we're looking at in caves is dark light. So you're in a cave where you cannot see the hand in front of your face, but there is plenty of light in there. It just happens to be in the near infrared. So I know my graduate student, Asuru, is online. He's the most enthusiastic astrobiologist I've ever met. This is his research project, and he's trying to understand the ecology of these systems. One of the systems that we've been working on is in Borneo. I did get rid of most of our data to fit within the 10-minute time frame. But this is an image from inside an entrance in Borneo. And again, it looks pretty bright in there, but this would be the twilight zone. It's just artificially lit with flash bulbs so that it makes it look bright in there. But if you look on the surface of this rock, you see all these kind of pinnacles pointing towards the daylight. This is a much more obvious picture from here. This only happens in the twilight zone. When we look at the community in there, there are a lot of chlorophyll D and a lot of chlorophyll F species. There are also a lot of species within the cyanobacteria and the proteobacteria, the chlorofexide that we believe may be produced in bacterial chlorophylls. But these pinnacles are really, really distinct, and you only see them in this kind of red shifted light within these caves. And Borneo is pretty warm. These caves are about 27 degrees. Asuru came up with a method for quickly looking for the presence of chlorophylls in these samples. So he takes the samples and then he can actually use EEM to look for the presence of the chlorophylls. And Chris was kind enough to give us some chlorophyll F and D producing strains. So we can actually use this to determine the presence of these chlorophylls quite quickly. So in addition to the 16S work to show that you had chlorophyll D and F producing species in this environment, we also were able to see the presence of the pigments. What's really interesting is this is in Borneo, so it's in a tropical region. There's a lot of energy. In 2019, I was invited to Greenland on an expedition, and we actually went, this is a map of the geology of Greenland, and this is a carbonate up here. And we were invited to go up there and look for caves in that portion of Greenland, one of the most remote places on Earth. I think at one point we were the first most northern people on the planet. This is 1,000 miles from the nearest settlement that only has 6 people in it. And then this is the carbonates. And I was invited by Gina Mosley. She has a START grant from the University of Innsbruck looking at carbonates to do paleoclimate. It's the most Mars-like landscape you've been to. It is a dry valley. The ice cap is way over here in the distance. And I believe it gets less than 10 millimeters of rain every year. So really, really Mars-like conditions, but it is full of caves, and the caves are very cold. This is me in one of the caves. The ambient temperature in this cave was -9 degrees centigrade. So this is hoarfrost just from air moving around. And it contained those same pinnacles, not as obvious here, a little bit more obvious here. And again, we could see great green pigments. So we're seeing this kind of photosynthetic activity, not only under near infrared conditions, but also at -9 degrees centigrade. So relevance to astrobiology. This is where we are. This is kind of a plot of all the stars in the galaxy, and the sun sits here as far as temperature, luminosity, and we are down here seeing a lot of active microbial activity in terms of luminosity down at 0.03 micromoles, and also we are redshifted. So there's a whole bunch of the sky that we could maybe examine and determine if there's life. Again, this is what, if you were to look at the light output from an M-type star versus a G-type star, such as our own, you see a shift. We see the same shift. You go from the entrance to deeper in a cave. What we've done is worked with some folks at NASA Marshall. they have a kind of backpack cave mapping device, a mobile device, so they can go in and 3D model the cave. Then I had the idea that maybe we could model how light goes into the cave and how it gets reflected around. And then you could actually pinpoint places in the cave that would represent the amount of luminosity and wavelengths for maybe our sun, what the light would be on Mars, what it would be in the TRAPPIST system, and then what would be the limit. And it's really easy because you just go in and is it green or is it not green? And then you're looking at your limits for the amount of luminosity, so the amount of light getting there, but also the wavelength. And then the folks at NASA were able to start doing that. And this is just the green light beams coming in, hitting the cave entrance and bouncing around. And if you look over here, you're completely in the dark zone. This is one wavelength of light and how it would bounce around. And you see it's just zipping around all over there in the dark. And then just a final picture. This is one of the caves we're working on in Borneo. To give you an idea of scale, that wall there is about 400 feet to this point right here. When I was in Norm's lab, he told me that the 16S ribotyping happened because he needed some mRNA molecules, and Tom Brock told him to go to Yellowstone because you can get a bucket full of biomass and extract all the messenger RNA you want. But then he was like, well, how are we going to figure out what's there? And Norm was like, well, just sequence the 16S. You can get bucketfuls of pigments and cells from these walls. We aren't going to be limited by how long it takes to grow these things in the lab because these cave entrances are stable. This one's been stable for over 300,000 years. So you can actually look at these ecosystems, pull the pigments and analyze them without being limited by depending on growth in the lab. So I think that's me on time. I had a conclusion slide and totally forgot to do collaborators, but there's a lot of people in this room I'm collaborating with, and I've read a lot of papers from everybody else in this room. So thank you.
So the mic disappeared, so I'll talk into this. That was a fabulous talk. Do we have any questions? Go ahead, Nancy, if you want to.
So those unusual, I don't know, stalactite-like structures that you see facing the light, have you looked to see if any of those cyanobacteria make intracellular calcium carbonates?
Well, we have a whole calcium carbonate. Calcium is toxic to microorganisms above about 80 nanomolar because it starts replacing other cations in the cell. So they have a very elaborate system for pumping out calcium ions. And eventually you hit a thermodynamic gradient and you can't do that anymore. So they'll, in caves, precipitate a lot of calcium carbonate.
Inside their cell?
Outside.
Outside,
All
okay. Some will
outside.
do it inside.
Look, cave bugs outside. But what we do is when we look at those pinnacles, and those were pretty small, they're up to a meter long in these caves. They're just enormous. They actually look like terracing. So you can imagine a very steep, high hillside in China and how it's all terraced. It's actually terraced like that. So the cyanobacteria, they're weathering the surface, but there's also some precipitation. So you end up with this, and it's almost like you have a point that points like this, but when you look at it going down, you've kind of terraced it so they can still get that profile towards the light. But those pinnacles always point exactly towards the light's coming into the cave.
We will restricting the schedule later.
Hi, I'll make it quick. So two things that jump to mind. One is a Mars relevance. So people talk about looking in caves or at least overhanging areas too because of the high UV flux. So not only would you be shielded from that, but you would get preferential because of the redshift. You would be getting the right light, right? So
yeah.
This would apply in a Mars cave, especially.
I did bring that up, and
I'm so sorry.
No. I did bring it up in talk. I brought it up with people, and I got completely poo-pooed because if it's open to the atmosphere, it's going to be so desiccated. The argument is maybe in your subsurface you can have some kind of saturation, but the idea is it just too desiccated.
I'm not arguing that it's happening on Mars. I'm just saying that is relevant.
Yeah, I did propose it, and then somebody said, well, why don't you start looking at N-type stars? That's much more relevant. And then this whole thing is about that. Yeah.
The only other thing I would point out is there are descriptions of photosynthetic mats that are below the surface, and they've been linked to this scattering effect on the scale of individual mineral grains, where you get a light intensity that's not right at the surface.
Yeah, we noticed that in the entrance, right? So in the entrance, you have that kind of black from the melanin that they're producing because it's just too intense light. And then you break that off and you'll actually see 2 millimeters down within the carbonate, you'll get that big line of green as well. So, we go from one extreme where they're using the rock to kind of diffuse the light as a sunscreen to where the light itself is actually allowing them to grow inside.
All right. Because I've done a poor job as a session chair, we've run over. So the boss said that we're going to come back at 2:40. We'll still take a break.
Thank you. The whole schedule will be shifted by 15 minutes. And we'll just make the breakouts just a little shorter.
I'm so glad you guys are doing that ancient enzyme reconstruction.
Yeah.
Something I really wanted to do before I retired, but it never happened.
We've done a little transformations already, so we're excited to see if they grow. Yeah. We've got a number of plans on how to address it.
My idea has always been that the ancient enzyme was non-selective. Right. Would you produce both rates?
Yeah. The problem is there's no such thing that we know of called bacterial protochlorophyllite. And that
Hi.
would be.
Can I use, people are struggling to see their slides. Can I use the
Yeah.
slide advance? Would it be okay to use a clicker or does?
I think this works. Let's see. It
I'm
was.
just wondering about using the clicker so I can stand
Yeah,
on the
it's
side.
not Yeah, we asked. We're going to ask them to move this over to there and get rid of those tables later. Sorry about
Can we
that.
just slide everything?
I
think
It's actually really good for you to practice because this is kind of a
Yeah, that's what I was saying.
Yeah.
And I was counting on
Okay.
being able to see the words. Well, I can see them on
Yeah.
the slides themselves. so
Yeah,
that
no,
helps.
it's a little.
That's all right. Don't sweat it. I'll just look at the slide.
Okay, anyway, they're supposed to come help us move it
And there
over.
is a
Yeah.
pointer that works.
Yeah.
That's your pointer.
So, let's see.
I would say not. People just don't.
Oops.
People just don't think about that kind of stuff. So the only pressure stuff, because like photosynthetic.
Did you upload your slides here?
I did. It's
Oh.
I didn't follow your naming nomenclature, so it's
oh,
under
okay.
P. It's photosynthesis lines. It's,
Oh, okay.
I'm
Phew.
out of-
All right.
out of
Okay.
color. That was my bad.
Okay. All right. I found it. There you are. Oh. Yeah, we asked
Yeah, I
them
was
to.
just
We
standing
asked.
here
We asked
thinking
them
it's
to.
blowing.
Yeah,
Just
it's blowing
thinking
on
the
us over
same
here.
thing.
Yeah.
Is it blowing?
Yeah, it's blowing on us.
They do photosynthesis just like the ones.
Okay.
To be honest, I really think that they came from up here. Not a lot of people like to imagine that being the primordial thing.
No, I would imagine.
Are these maybe the people who are here to move things?
Oh, yeah. Okay. All right. Can we move this? Because people standing here cannot see their screen.
Sure, can we maybe move this?
Yeah, we can get rid of. We
table
can completely get rid of this.
And then just move
Yeah.
this podium there. and-
Yeah. Oh, that'd be great. Thank you. Thank you.
This is not just for me.
Oh, you know, it's for everyone. Yeah, no, yeah.
It's really a pleasure.
Yeah. Yeah.
Thank you.
Sure. And you'll be wearing this, Tim, for sound.
What's that?
You'll be putting.
Yeah. That's
All right.
So we've seen how that cyanobacteria will leach metabolites out of those soils. And we're kind of quantifying what certain cyanobacteria that do that, what exactly they take from their environment, that sort of thing. But obviously there are other, I like to think of it as resources or like even just like physical parameters that people haven't looked into very much of.
Yeah, a lot more now. Surface models.
I should just. Oh, you're wearing a scarf in here. Yeah, and it just went on the afternoon.
Outside is
Yeah.
much warmer.
Maybe because we have the door open and it thinks it's warmer. Maybe we. Yeah.
Well, maybe at some point I'll like pick your brain about some. I don't want to say low hanging fruit, but some easy types of.
Yeah, right.
That we could start doing too.
Should I just trust and then
We're a little bit of a.
assume the slide works, or do I need to test it?
Oh,
Yeah.
most people work on, a couple
No,
of
it's
songs,
on speaker.
so
we'll just swap
50 different yeah.
I need to defrost. So using this is better, actually.
So
As a pointer.
You can use this for a pointer. I don't know if you can see here. This is a pointer here.
But for the, maybe it's easier to point with this. What do you think?
This is a pointer. I think this is.
I can use this one.
Oh,
on the screen.
I don't know if it doesn't show up on there.
It doesn't show up? Oh, really?
This way. Turn around this way, and then if you press the red button, there.
Uh-huh.
There.
Okay.
Okay. So that's
Sure. Okay, great. And slide change should be this one or this one as well? Let's try.
it was working once, but now the clicker doesn't seem to be doing it. So just use the arrows to.
So
Sorry, I'm in the wrong. Here. There. So when we're in the PowerPoint, then I think.
I see.
I think you can.
I think that's kind of
Doesn't work. Okay, so just use the arrows.
Okay.
Yeah.
Got it.
There's like six things.
So there's certain like people will look at microbial math the heliobacteria have the simplest reaction center. It's basically just 2 core polypeptides, and that's it. You kind of think that would look like the most ancestral one, or at least it's like the closest thing. Those grow in anoxygenic, like rice paddies, right? So I would have no idea if that would be in some ways like similar to what because you're not right.
Is this the final spot?
Huh?
Is this where they're leaving?
I wonder where they went.
Because this is
This
is even worse.
I'm calling them
Okay.
Coming back.
They're coming back. They were
Hi,
pretty.
are you gonna move? Oh, they're coming back. Okay, I'll move this.
Earth environment.
I'll just hold this up.
Like, yeah, I don't know.
Yeah, I mean, so a lot of people have
Yeah, so here.
astronomers.
I'll hold it for you. Use this in my hands.
Probably don't put that on.
Yeah. Yeah, right up there. Avi, we're gonna move this over there.
That was the original.
Okay, you can turn it. Yeah, at an angle. At an angle that way, so that they can look at not too much, not 90 degrees, but 45. How's that? 45. Okay, so they can look at the room, and then they can look at people. Okay, and we you got tape. Maybe tape this down too.
So yeah, so
Thank you. It's much better.
Like I
This is this.
Yeah, like I wonder if you could. I was just kind of forced to write this like AI, right? And basically, you can take a lot of that big data and start using like machine learning to actually like, say highest and lowest probabilities and things like that. And if there were a repository for those and physical parameters for different places on Earth, we could imagine like trying to kind of match them in a big data.
I don't someone feel like folks that I don't know, just
Yeah.
I'm sure it's out there. At least in some parts.
Yeah, I mean, I know they have like the oceans or whatever project, and that samples like has a lot of that.
Okay.
Yeah.
And it just advances the standard way with the arrows?
Yeah, use the arrows.
Better to use the up down, or does it matter which ones you use?
Oh, I use the side to side.
You use the side?
Left, right.
Yeah, that works.
People say,
That's easier.
and some
Okay.
they're all red light, and they're all like super low pressure, and they're all not gonna have X, Y, Z. That's why they're not like There has to be limits. And that's what, obviously, push the limits to a certain point. At a certain point in the cave with not enough light, so it's a beautiful real world test. Yeah. Observatory research surveys and all planets we might find. But at least maybe we'll be able to say this is the range of these
Yeah. You know who would know a lot is sort of the microbial ecologist. And we have a lot of ties with that because we like resources and microbes. I'll bring you up with a first operator and see if they have any comments. But it's, yeah, I think there's something.
There's a big one.
Yeah.
But I'm sure.
Yeah.
I'm sorry, because it's just it's turned into camps about whether you can because of the stellar effects. It's great to see still a community here who is interested in M stars.
You mean we don't know better?
I know. I think it's biases. I'm an M star hugger myself.
Okay.
I think there's so much we have now.
Yeah.
Yeah, well, we're pushing outwards, unlike G stars. And more sort of planets, large distances. M stars are working their way outward. But that means we're seeing the most sandblasted, highly irradiated population. It's making people impressed because they're not seeing atmospheres. But it's like you can see a bunch of Mercuries, and now you're going to judge the whole planetary system. Anyway, we're not there yet. Exactly. We need to, but it takes a next generation WST copy of
I dance on the side of all of these speeds, it's relevant. But I always look at the species. Right. Because it's between what's not possible and what's not possible. Yes,
absolutely.
1,000 years from
Absolutely.
technology.
And what's amazing about how NASA works, which I love, is if you can motivate how important it is, they'll figure out a way to do it. Exactly. So if you say there is no other way to absolutely confirm 100% of this, I'm saying, okay, that's what we need to do.
That's what should always be, unless it's a physical measurement, that's just not possible. We're not going to go there. Sure.
British. That one is called out. Yeah, so it's fun. It's a fun time because it used to be that, right? With techno-signatures, people would say, don't waste your career, whatever. We're in this time where people are more.
Which is interesting because it coincides, collides.
I know. That's what's so whiplash. It's the whiplash. You talk to one group of one population, go, we love this. We'll put half of our GDP towards this. You talk to the other one, yeah. Focus on
Administration to administration,
yeah.
groups within the president.
All over. Right? Even speaking to, say, a public audience, right? Half the audience will be like total, enthusiasts, and the other half will be like, well, tell me why it matters.
Totally. I mean, I think you should have your feet held to the fire. Right? It's
Of course.
and there usually is.
I just feel like it's more and more polarized.
What I can tell you here, that's generally.
Yes.
I didn't bring a poster. And that's easy.
Yeah.
That's easy.
Yeah.
Do I need to That's obvious. I can't believe how long certain people have your own work. But the exoplanet is the opposite. Anything like this, that's like it's not just
Yeah. Or if you look at, say, DPS or some of the other ones, like, something like that.
Okay,
How come
that's
nobody's been using this? It doesn't work?
I was, but I think it's really hard to see. Do you want a green one?
Sure, you have one?
Yeah, it'll be way brighter too. I should have used it. So yours are on there, so they should be able to just double click them and it'll come up. So I think you're good.
Awesome.
Yeah.
I think it will have to mature.
Yeah, that's way better.
I'm ashamed that I
All right, I'm gonna get my pointer set.
I'm usually pretty adamant about
Oh, yeah, it's great to see you. I knew we were friends with each other.
Yeah.
So we'll have
I'll just leave it here, Chris.
Yeah.
Is this-
Yeah.
Especially I'd like to.
Okay, now I'm gonna get some tea. Good luck. What number?
Are you
Second to last, I
are
think.
you going to upside?
It was supposed to be 3:45,
I
but
have a
it's.
similar meeting, also I need to leave the area. I think if possible, I'd
We can find a time.
Yes, yes.
That's fine. Yes, that's great. It's great to see you.
It's-
Yeah.
Chris,
That was her birthday.
are you, I think you're session chair? Yeah, you're session chair. Yeah, I know.
Okay.
Y'all set? Yeah.
Neighbor, I never gave this to you. I'm sorry.
So I'll give you the 5-minute warning and
Yeah.
maybe a 2-minute or so.
Definitely fun. Cool. Start talking faster. That's it.
We're gonna start in 3 minutes. Is that what you said?
No, you can start now.
Oh, okay. Everyone, we're gonna get started now. welcome to the next section. It is called Planetary and Biosphere Coevolution, Geochemical Context. And first up is Tim Lyons. Tim, you're all set.
I am all set. Okay. Thanks so much. Okay, well, thanks. Thanks for the invitation. It's really a delight to be here, especially amongst such a diverse crowd. I'm learning a lot, and I really appreciate that. If it hasn't been obvious already from my questions, I'm trained initially as a geologist, and then as a geochemist and a biogeochemist and an astrobiologist. And I've led a group now for about 10 years where we've turned decades of earlier studies towards the questions of biosignature development, templates models using Earth and what we call alternative Earths. Earth has actually been many different planets if you think about its history. And using those different chapters, those different slices to inform atmospheric evolution and think about how we can look for these things, in particular around exoplanets. It's a brief history, but it's not a brief title, and I apologize for that. So we'll move past that. And if you know me at all, you know that I've spent a lot of time thinking about oxygen. And so we're going to talk quite a bit about actual records. Some are very robust, some are less robust, but it's sort of the state of art of what we know right now about Earth's oxygenation history. And I think it in particular informs a lot of things about, this is not advancing. Sometimes you have to do it once on the screen to wake it up.
No, it has to go into PowerPoint. Yeah.
Yep, thanks so much. And so I decided that I was going to sort of break this down into Earth lessons, as I said, and I try not to be too Earth-centric in this perspective, but again, Earth has been a lot of things, and in a process sense, in a product sense, which are really what the biosignatures are, it teaches us a lot, and a lot of things that will be universals one way or another. So we'll talk about records, timelines, and a planetary systems perspective. That's something I'm very much into. I will give you the $1 million punchline right at the beginning that most of what this is about is nutrient availability in terms of the biosignatures and where they are and why they are and how detectable they are. And Earth has taught us that lesson in space. So we'll talk about the onset of the signal, oxygenesis in terms of atmospheric signals and pigments, the timeline, the dynamics. It turns out our oxygenation history has been extremely dynamic. The spatial landscape controls nutrients. Also, the importance of continents. Earth has been a planet that has not had continents, and then, as I said earlier, continents that were underwater, and then only over the past 3.5 billion years has it had emerging continents. And then the competing reactions that one set of processes producing oxygen and the others consuming it at the same time. And the great oxidation event that I'll talk about is really a product of that shift of balance between consumption to production and buildup. It's not probably a biological step, and you'll see why I'm saying that. Implications for other microbial pathways, including other potential biosignatures, and implications for remote detection. And so the classic view of Earth's oxygenation, as many of you will know, is that there was a great oxidation event at about 2.3 billion years ago. And then something that's called the Neoproterozoic oxidation or oxidation event at around 800 is actually ironically less well constrained, even though it's a lot younger, because this is so well constrained by a series historically of geological tracers, like the presence versus absence of detrital pyrite minerals that degrade under oxid conditions. There's a long list of things that people used long before we had geochemistry. Now this is fingerprinted extremely well with the most magical tracer, and that's mass-independent sulfur fractionation. So it's the presence of that before the GOE and the essentially complete disappearance of mass-independent behavior of sulfur through atmospheric processes after the GOE. And so Earth's oxygenation history, as I've already said, it was a really dynamic story. And another thing that's really critical to understand is that the Great Oxidation Event, according to pretty much everyone, if not everyone at this point, the Great Oxidation Event occurred hundreds of millions of years after the beginnings of oxygenic photosynthesis. And that speaks to this idea of its production versus the sink terms. And so for the longest time, we wanted to be able to fingerprint in a geochemical sense the beginnings of oxygenesis. And there was a landmark paper published in 1999 by Brox et al. That argued for the presence of hopanes and steranes, so cyanobacterial biomarkers and eukaryotic biomarkers from 2.7 billion years ago in a core collected in northwestern Australia that has subsequently been revisited, and all of that is contamination. So this no longer stands as any kind of golden spike landmark for the beginnings of oxygenesis. So we turn to other things, and some people like Tanya and others turn to fossil evidence, conical stromatolites that speak to phototactic behavior, cyanobacterial production of oxygen, perhaps in this paper argued at 2.7 billion years ago, bottom line, hundreds of millions of years before the traditional view of the great oxidation event. And that process is fingerprinted through fossil bubbles and contorted laminae related to oxygen migration within those structures. There is also a wealth of genomic data, and Greg is here, and I'm not going to pretend to even speak to what he and others have done so well. But the bottom line is that most of the genomic data, the molecular clocks, speak to the development of cyanobacterial oxygen production 300-ish, long before the GOE, again, hundreds of millions of years before the canonical view of the GOE. What we really do is inorganic geochemistry, and it's ironic that that would be, in my mind, one of the most convincing arguments, not really for, what? I'm sorry. You think cyanos developed right at the GOE? I know there are some of you. And it is ironic because everyone wants to find the organic biomarker. We will never find that in Archean rocks. They are just too buggered. We will not ever find a convincing organic biomarker in those rocks. And so we are left with inorganic tracers. And one of the ones that is my favorite, partly because it came out of my group when Noah Planaski was my student, is looking at rocks that are about 3 billion years old from South Africa. South Africa and Western Australia are really the best places to go for rocks of this age to do these kinds of things. And the long story short on this is that there's this relationship that when manganese oxides absorb molybdenum, there's a very strong negative fractionation. And so if you look at this paper, what you'll see is we made the argument in these 3 billion year old iron formations that the more manganese there was, the lighter the molybdenum isotope signal, which spoke to the presence of manganese oxides. So implicit in this argument is that manganese oxidation requires O2. And so I'll speak a little bit more to that in a second. But as the amount of manganese oxide goes up, the isotopic value of molybdenum goes down, again, because of this very strong negative fractionation onto the manganese oxides. And this proxy is used in a lot of different ways throughout Earth history. There was a challenge to this, and Greg was part of this paper, where they argued that there could be an anoxygenic photosynthetic pathway that would produce manganese oxides without the presence of O2. Now, this is a pathway that people have been looking for decades and decades, and some really elegant work that came out of MIT suggested that they may have found that. I'm not going to argue that. I would love to do that in discussions and get involved with Greg on the current status of all that argument. But in these kinds of tennis matches, the ball comes bouncing back to you when there's an argument for manganese oxides that are fractionating molybdenum that have nothing to do with O2. What you have to argue with, which I think is the reality, is that manganese oxides, if they form through a non-oxygen related process, would never be buried because of the rapid dissolution of manganese oxides in an iron-rich or hydrogen sulfide-rich ocean. So it's not just about scavenging the molybdenum, it's about delivering it to the sediments and preserving the signal. And we think that would be very hard to do without surface ocean oxygen. So then it begs the question of, if oxygen production is beginning so early relative to the GOE, what delayed the GOE? And I've alluded to this already. There are many different models that talk about reductant fluxes that consume oxygen that would change over time, hydrogen availability, subaerial volcanism that changed the redox structure of sulfur emissions. There are arguments about changing continental composition that Nikki alluded to before, mafic continents more effectively scavenging oxygen than felsic continents. There are even arguments for the GOE being related to the changing day length. So there are a lot of arguments, and most of them work in the same direction, so it's probably not one single thing. What we see when we look at the proximity of the GOE in time is that we think pretty convincingly you see evidence for false starts, occurrences of oxygenation, not large necessarily, not long-lived necessarily. You still get mass-independent sulfur fractionation at this one that we've argued for, the famous WIF at 2.5 billion years ago. But it was an early warning for what we think, and many people think now, was a really oscillatory transition from one state, no oxygen, to the state that we entered into the Proterozoic and, of course, into the Phanerozoic. And this is a purposely busy figure because what it shows are data from many different places, a number of different intervals that go across the GOE. And it uses traditional approaches like the detrital pyrite. It uses mass-independent sulfur fractionation. Long story short, what these data argue, when you correlate as best as you can with the glacial deposits and other approaches, but in particular the glacials, is that there is this really oscillatory process where oxygen rose, then fell, then rose, then fell. And I used to be skeptical about that, but now I'm convinced. And so now using many different kinds of geochemical tracers that speak to the surface ocean, to the atmosphere, to the deep ocean, we started to put together a new oxygen curve that builds from the one that I published in 2014, which is widely cited for better or worse. And so here is the new one, and I promise this will be my last oxygen curve. But you see this increase from very low to still very low oxygen. This is constrained unequivocally by the presence of mass-independent sulfur fractionation. And then you see hints of whiffs, some stronger than others. There's the 2.5, and then this oscillatory behavior. And then at the tail end of the GOE, there's the suggestion of an oxygen overshoot during a very strong, long-lived positive carbon isotope excursion. And then the idea is that oxygen relaxed to some lower level and then rose again at the Neoproterozoic oxygenation event. So we can now start to map microbial evolution on top of this. And so we've taken a number of different cycles. This is just one for nitrogen, where we actually have color-coded when we think these things developed and how they fit within the environmental contexts that would have been present at that time. For example, nitrification appearing when we would have had oxygen in those oases in the surface waters. And so there are a lot of things that fall out of this. There's the GOE. You can see a lot changing at the GOE when oxygen for the first time develops accumulates irreversibly within the atmosphere. And there's the NOE. And so once you have oxygen, not just oscillatory, but staying there persistently, then there's a lot of potential for development of aerobic organisms. But interestingly, you see up here, this is just one example, that nitrogen fixation seems to predate the denitrification. So what you can imagine, and nitrification. So as oxygen is first starting to accumulate in a meaningful way, you can nitrify and denitrify. And that's the first time that you're actually returning N2 to the atmosphere. And you're stripping a lot out through earlier N2 fixations. So there have been arguments that total atmospheric pressure was lower at this time, and that fixed nitrogen availability may have been a challenge at that time. So the bottom line is that oxygen rose, but it didn't go from nothing to what we have today. It oscillated, it rose dramatically and fell, and it stayed in the famous boring billion between roughly 2 to 1 billion years ago at some low, maybe 1 to 10% of present atmospheric levels. And so you have to ask yourself, beyond these reductant fluxes, what else is doing that? And of course, it's going to be the nutrients. So much of this is about nutrients. So we have throttled biospheric oxygenation, particularly in the mid-Proterozoic, as related to limited nutrient availability, and follow the phosphorus most importantly, but also important challenges related to nitrogen through the nitrification, denitrification cycle, because you have a lot of oxic water in the surface sitting on top of anoxic water. So it's easy to nitrify, denitrify, and then lose that through as a channel of N2 back into the atmosphere. And under those anoxic conditions, it's really easy to keep enzymatic metals at low levels, like molybdenum and potentially vanadium. But we and others have argued that molybdenum scarcity may have throttled nitrogenase and some ability at high levels to fix nitrogen, although there's a lot of work going on with that now. And that's not my favorite model anymore because of this recent work, but it has to be part of the story. So it's going to be, these nitrogen stories are going to be the ultimate control on biosignature distributions and magnitudes. And so what was going on with phosphorus? Well, the ocean at that time, the deep ocean, was anoxic probably until about 400 million years ago. And rather than being sulfur rich, which Don Canfield suggested in 1998, a sulfidic Black Sea type ocean, it was almost certainly dominated by dissolved iron. And so dissolved iron has a deleterious relationship with phosphorus availability. As it oxidizes through photoferrotrophy, for example, it scavenges lots of phosphate out. You can get a trap at the chemocline in a water column like you see in the Black Sea. Vivianite, which is an iron phosphate mineral, can form. And so there are a number of different things that conspire to keep phosphorus limitation persistent for a long period of time. And so models like Monte Carlo simulations that suggested that net biospheric oxygen production was maybe 25% of the present value in the mid-Proterozoic. And a paper that came out of Harvard said that it maybe was 10 to 1% or less of present production and oxygen released through cyanobacterial activity. So there are, again, a lot of different things that point to the controls on oxygen, and central to that, and this is going to be true on any exoplanet as well, is what nutrient load do you have and how do you keep recycling that, whether it's nitrogen and phosphorus or phosphorus. And so another thing that has come out of our work recently is that there's a fundamental change in the nature of mafic rocks as we have decreasing degrees of mantle partial melting, the Earth is cooling, and so the mafic rocks that are produced become progressively enriched in phosphorus, which helps for the nutrient limitations, and nickel decreases, which hurts enzymatically for methanogenesis. So an Archean rich in methane makes sense because of nickel, increasing oxygenation because of increasing weathering of, in particular, large igneous provinces that have phosphate, that help. So this offsets a bit of those lost channels that you have for phosphorus in the ocean. And so this is what we think the ocean looked like for long periods of time, probably starting in the late Archean. It was mostly ferruginous, sulfidic on productive margins where you had enough organic matter around to drive sulfate reduction, and you had enough sulfate through oxidative weathering of the continents. And so the first thing that jumps out of this is that you're going to have an oxic layer in productive areas where there's lots of cyanobacterial activity, and that sulfide would be at depth. So if you're looking for purple sulfur pigments or green sulfur bacterial pigments, you might not see them because that plate of activity would be driven deeper into the waters. And so we have started modeling what oxygen availability would be like in the ocean, and this speaks to distributions of pigments if you're interested in looking for chlorophyll A, for example. And so not surprisingly, the oxygen, the earliest increases in oxygen in the surface ocean were in areas where you had a lot of upwelling, divergent zones along the equator, or the intertropical convergence zone more correctly, along the western margins of continents and at high latitudes. And only when you get to fairly high levels of oxygen, 10% of the present atmospheric level, do you see broader ranges of oxygen in the surface ocean, because now they start to be controlled by the atmosphere and exchange with the atmosphere, while these oxygen distributions are really about in situ production in highly productive areas. Think cyanobacteria blooming, thriving, releasing oxygen locally. And so it's not surprising that these maps look like a chlorophyll A distribution in the modern ocean. And so we started to take all of this and think increasingly about what it might mean if we were trying to look at Earth from a distant world long ago, and what kind of models we could develop as we think about biosignatures around exoplanets. And so we submitted a CAN7 NAI proposal that was funded, and then from there we built an ICAR that was funded about 5 years ago. And it has been really about how to build and sustain a detectable biosphere. And unlike the NAI proposal, this one had a very strong presence of exoplanet people, including Eddie and Stephen Kane, who's not here. And so we really did and have developed ideas, models from this early Earth perspective, as you've been hearing throughout the meeting. One of the things that we learned right away is that the Earth is a great example of a false negative. If we are correct that oxygen first accumulated as oases, giving us that molybdenum isotope signal 3 billion years ago, while the atmosphere was extremely low with mass-independent sulfur fractionation, that it's possible that detectable oxygen in the atmosphere may have taken 2 billion years relative to its first production around 3 plus or minus billion years ago. So we may have had a false negative of surface ocean oxygen, again, for 2 billion years, where aerobic metabolisms are developing, intense diversification is occurring, but there's no oxygen expression of that in the atmosphere. Again, the sulfur emit signal that you have for the Archean sets oxygen levels at about 10 to the minus fifth to 10 to the minus seventh of the present atmospheric level, essentially absent, completely undetectable. And so we're thinking within that framework about what methane biosignatures might look like as well. It is always assumed to be high in the Archean atmosphere. It's not in the Hadean because of early core formation, and that's a whole nother talk. And so there are papers now, arguments recently that suggest impact-related delivery of reductant might have given us transient events of methane accumulation in the atmosphere. But the Hadean greenhouse is mostly set on CO2, which has really important implications for pH. pH is perhaps in the 6 range, which has really strong implications for proton gradients if you're in origin of life sense, and presence or absence of borate, as people argue for the for the RNA world synthesis scenarios. And so, and so once we get to the great oxidation event and oxygen accumulates and methane destabilizes, it's mostly a CO2 world going forward as well. But the Archean has always been linked to very high methane. Biological methane production, and this is work with Greg and my student Sally, who's here, 3.8, perhaps 3.5 billion years ago. The ocean composition certainly favored it by having low oxygen and low sulfate, so you're not losing methane that's produced through anaerobic oxidation of methane. You can lose it through iron-based AOM, and that's something that we're exploring. And then as we go into the Proterozoic, we have increasing O2 and increasing sulfate, which challenge both the production and the preservation of methane. And of course, we wrestle with biotic and abiotic records. But one possibility is that the Archean look like this, where we have hazy atmospheres. We don't really know. There are some sulfur data that suggest the presence of hazes, but it's really hard to know for sure. And the green ocean that you're going to be hearing a lot more about tomorrow, I think. And people have argued for purple oceans, potentially on productive margins related to purple sulfur bacteria. And so the question then is, how does this all fall out if you were looking at Earth and trying to think about its detectability of biosignature gases? And so what our results argue is that even if methane wasn't as high as people have speculated, it was certainly high and almost certainly detectable in the Archean, but O2 not at all. In the mid-Proterozoic, contrary to traditional views, O2 was probably too low as well, and we've argued for ozone because of its potential enhanced detectability relative to the O2. And we have also argued that methane, and this is a controversial idea, was probably below detection. Because you're increasing oxygen, you're increasing sulfate, again, affecting production and preservation of the methane. And oxygen still may have been low enough that the ozone shielding may not have been effective enough to reduce sufficiently the photochemical destruction of methane. So we have published the idea that not only would you not have the classic methane O2 disequilibrium, but you may not have seen either of those gases for 1 billion plus years in the mid-Proterozoic. And then of course in the Phanerozoic you have O2, but methane below detection almost certainly, depending on your instrument. And so I'll sort of end on something that I think poses particular challenges, and that's the idea of what water worlds would be like for those of us who are trying to look for life on them. And we have a water world, right? The early Earth almost certainly was, and I'll make more of a point of this in a second, but we almost certainly did not have traditional plate tectonics until about 3.5 billion years ago. Early continents, they came in the Archean, so we wouldn't have had continents. As I've said already, they would not have been emergent above sea level. And so we have to ask really fundamental questions about early Earth, but also for water worlds that we might be looking at in distant places. How do we cycle nutrients? You can fix nitrogen potentially on an exoplanet, but it's really hard to recycle phosphorus without having either plate tectonics, where you keep recycling it through uplift weathering cycles, or through seafloor weathering, which is a popular idea now. But even seafloor weathering requires resurfacing. You have to have new seafloor to weather to get a new load of phosphorus, which implies seafloor spreading or plume events, but it implies some kind of tectonic deep earth process that's giving us a fresh supply of phosphorus at the surface while it's being sequestered through biological processes. So resurfacing may be essential. Are there implicit assumptions about tectonics? If you're talking to me about a water world and sustained life on it, the first question I'm going to ask you is, how do you explain the nutrient balance? And I think that's a really critical question. And Vicky and I and many other people talk about ways of trying to infer the presence of these kinds of tectonic recycling controls. And so Jun Korinaga has been really a pioneer in all of this, in particular about understanding the onset of plate tectonics and the emergence of continents above sea level, which becomes really, really important for origin of life scenarios. If you need wet-dry cycles, for weathering, for the silicate weathering feedback that modulates CO2. And if you look at this figure, and many people have come up with compilations, but the agreement on when continents first appeared and when they became emergent is not very strong. And so this is early continent formation and then a plateauing. Other people have argued for something through here. There is not strong agreement. I will go on record as saying that we don't really know when continents formed, when they grew, when they became emergent. And that's a really important question. We're getting better and better at figuring that out. And so to go back to the initial questions, the onset of signal oxygenesis, O2 pigments early, I would put it based on just our geochemical data that there's evidence for accumulation, at least in the surface ocean, going back about 3 billion years ago. The timeline, dynamics, landscapes of oxygenation, incredibly complex, right? Because it's about production, it's about sinks, it's about controls on nutrients. And there we have that, tectonics, continents, and competing reactions, which again are drawing down the oxygen. They will vary strongly from planet to planet for sure. Implications for other microbial pathways, absolutely. Sulfate availability, for example, becomes important once oxygen rises. Sulfate generates sulfide. Sulfide becomes important for green and sulfur bacteria, which then generate potentially important pigment records. So these things are all deeply intertwined. And at the core of it, most of it is this early production and accumulation of oxygen. So implications for remote detection, sure, many challenges. I think the only message I would leave you with is that they're not insurmountable challenges. You have to take this from an entirely planetary system sense. You have to imagine all of these pieces. To do any one of these things in isolation has almost no meaning because they're all so deeply interconnected through really complex feedbacks. And that's why Vicky loves to have these complicated figures, because it's exactly what the world is, right? Those are all interrelated pieces. And if you change one, then you change a lot of other things. And so that's really what we're trying to do. Thanks.
One brief question.
Thanks. You mentioned the possibility that you could get phosphorus from seafloor weathering, but it seemed like it needed to be anoxic. So wouldn't that kind of like self-limit the ability of the water world to build up the O2 anyway?
You mean because of the sink for the phosphate once it's released?
Yeah.
Yeah.
You can't hear that.
Yeah, sorry about that. Yeah. May I hold the microphone just 1 second? Yes. So the point is that in a ferruginous ocean, which is what we call an iron-rich ocean, you'll be hearing more about those at the meeting, there is a really strong capacity to scavenge phosphate. I made that point, right? And so you can do it when there's oxygen in the surface ocean. And there would have been oxygen in the surface ocean, as I suggested, even from the late Archean all the way through to the present, with eventual deep ventilation by the Devonian. That's the best guess that it took that long. So you had tons of iron, but you had oxygen in the surface waters that could have driven that oxidation and the scavenging, like it does in the Black Sea. There's this amazing iron particle maximum that scavenges all the phosphate. It drops down, and you get this kind of pump that goes on in sediments as well. But there's also photoferrotrophy, right? And so you don't even need oxygen. If you have that anisotropic pathway, and that's an argument, controversial, sure, but it's a popular idea for how iron formations form, at least one of the ways that they form. So you have a way of getting oxides scavenged in phosphate. What I would say to your question is that in a world that's doing that, if you're also not putting new phosphate into the system, you're really screwed, right? And so it doesn't make the problem go away. It's just a way of keeping new phosphate delivery, fresh phosphate delivery into the ocean, where it can be scavenged. And also when the iron traps it, some of that iron falls and redissolves and releases it. It's just getting into the surface ocean. It's a really great question.
Okay, let's thank Tim one more time. Next is Junko Yano from LBNL.
Thank you so much. First, I want to thank Chris, Nancy, and Bob for inviting me to this very interesting workshop. So earlier in my career, I decided that whenever Bob asks something to me, I should say yes. I said yes without thinking, and today is my daughter's birthday. So I feel guilty, but it's okay. It comes every year, so. With that in background, so I'm going to be talking about oxygen evolving complex. It's related to already, there was a talk by Chris and also in a few talks. I've been working on photosynthetic system water oxidation reaction for a long time. And I'm coming back to this fascination of water research, how special water is and how thankful we are living in this planet. Why water is so special? It's a very simple structure, but there are partly charged protons and partly negatively charged oxygens. So this structure makes things special, and it creates this hydrogen bonding network. It can interact up to 4 waters. And that character creates a very complicated structures. For example, unique physical properties come from here, like high heat capacity, high heat of evaporation, and surface tensions. If we think about from the chemistry point of view, unique chemical properties, water is an effective solvent for wide range of substances. And also it can work both acid and bases. It means that it can donate protons or accept protons. For example, this hydrogen bonding network, the proton transfer can be very fast because each proton doesn't need to be changed. The location needs to be changed, but interaction between the water next to it can be just changed, and then proton transfer happens. So all these character properties makes water as a fundamental to life at every scale, starting from atomic scale up to the planetary scales. From photosynthesis point of view, water is the ultimate electron donor of life. So water splitting in photosynthetic system is the ultimate electron donor of life. In the anoxygenic photosynthetic system, early photosynthesis, the organisms couldn't use water, so it used hydrogen sulfide, ions, and hydrogen as an electron donor. The standard potential at pH 7 of these elements or materials, it's much easier to extract electron compared to water. So it doesn't need to have a high oxidative capabilities. And extracting electron from water is energetically quite demanding. As a consequence, the early photosynthesis depends on scarce geochemical electron donors and probably used longer wavelengths near IR of solar spectrum, which is lower in energy. This changed after the evolution of cyanobacteria. As we've been hearing from many talks, water oxidation reaction, which uses 4 photons, made our Earth in an oxygenic environment. So the catalytic center, which does this water oxidation reaction, as we already heard from Chris' talk, is the 4 manganese, 1 calcium cluster. These are oxo-bridged, and it has 4 water molecules ligated to it, W1, W2, and W3, and W4. Why manganese was chosen? So manganese 2+ was already available in the ocean, probably 120 micromolar level in the ancient ocean. And probably manganese oxidation chemistry was available already at the biochemical toolkit, like a manganese 2+, 3+ to 4+ redox cycle is often used in enzymes, multiple enzymes like manganese superoxide dismutase or manganese catalase. So all these are there. And maybe the earlier reaction center had the ability to oxidize manganese in it. So what does photosystem 2, current photosystem 2 does? So there's a reaction center chlorophyll right here, P680. This does a charge separation 4 times in order to do 1 cycle of water oxidation reaction, 2 water to create 1 oxygen or 2, 4 protons and 4 electrons. Here, manganese cluster. This cluster works like a charge accumulator during this 4 photochemical reaction at the P680, and it collects oxidizing equivalent between photochemical events. But in order to do this chemistry, P680 need to have enough oxidizing potential, probably 1.2 volt and above. And in between, there is a tyrosine Z, which has a slightly lower oxidizing potential, but high enough to oxidize manganese or advance S state of the water oxidation reaction in the manganese cluster. So these set needs to be evolved together in order to achieve water oxidation reaction. And this was already shown where oxygen evolving complex on photosystem 2 is located, so I don't need to go into the details. What we've been studying is the dynamic motion of this oxygen water oxidation reaction during the catalysis, but from the protein structural point of view, as well as sorry, it's falling. As well as catalyst point of view. So far, it's okay. So if we put the photosystem 2 or plant in a dark, it goes into the dark state. It's called S1 state. And then if we shine 1 laser flash, it goes to the S2 state. So S1 state is the oxidation state of 4 manganese in S1 state is manganese 3344. So starting from there, and it goes to 1-step oxidation of manganese to S2. And again, next photochemical reaction, S2 state goes to the S3 state. And then after that, S3 state goes all the way to the S0 state. This is the step that molecular oxygen is evolved. And this reaction cycle continues multiple times. Eventually, everything is mixed up. But this first, up to 3 flashes, we can synchronize the reaction very much. And this is the step we've been using for our structural studies. So what we want to do is to take a kind of snapshot at the various steps during this Koch cycle of the water oxidation reaction. To know the structure, there are multiple ways. Cryo-electron microscopy has been used quite widely. Or if it's a more traditional way, we go to synchrotron X-ray to do crystallography using a few single crystals. What we've been using right now in our group is to use X-ray free electron laser. This produces very narrow, ultra-fast X-ray pulses, femtosecond X-ray pulses. So using this, we do a shot-by-shot experiment at room temperature. So the advantage is that we can look at the structural dynamics or chemical dynamics using at the room temperature using this X-ray sources. The idea is this. For example, if we want to know how the bird flies, what do? We go capture a bird and freeze it, and we can do very detailed high-resolution structural studies by this way. This is not the way we took our research. We want to capture various moments at room temperature, first of all. But on top of it, we want to see, we want to take a snapshot during things are functioning. And so that's our approach. In our lab, we grow this cyanobacteria and then extract the photosystem 2 here, crystallize it. And these micron-sized crystals, about 20 to 40 micron-sized crystal, is what we've been using for our research. And we take these crystals to the facility, X-ray free electron laser facilities, to do a shot-by-shot experiment using this setup. So let's see. This we call tape drive, and this droplet, about 150 micrometer droplet, contains micron size of single crystals of photosystem 2. And then we do diffraction measurement many, many times, shot by shot. And at the end, we put the 10,000 of snapshot to create one picture of the structure. Simultaneously, though, we've been collecting X-ray emission spectroscopy right here. This is in order to know the state of the chemical state at the metal catalytic center. So then we can bridge the protein structure and the chemical state of the metal catalytic center together. Using these multiple lasers, we can take a snapshot at the various moments like this, and that's what we did. We took a snapshot at the various moments. I will go into a little bit of details. So S1 to S2 state structure, one manganese oxidation happens during this step. Structure of the cluster doesn't change much, just one oxidation state changes. But going from S2 to S3 state, this is the step that one of the water, potential substrate water, comes into the metal cluster. So we do see the intensity, additional intensity or density showing up. I'm showing here pink spherical shape. This new ligand is coming into the metal cluster. So that's what we see. And we think that during this process, calcium plays a very important role. So calcium is right here. And water will likely use this calcium ligand environment into coming into the manganese ligand site, ligation site right here. So that's what we see during the S2 to S3 transition. Going from S3 to S0 state, so this is the step that release of molecular oxygens and the catalytic center goes back to the original shape. We want to see which two oxygens are coming together and how the catalytic center recovers. Again, we took several snapshots. And I don't go into the details. What we think is that probably the O bond formation happens somewhere around here, 500 to 700 microsecond after the third flash. Likely location is here. And then in the time scale of 700 to 1200 microsecond, molecular oxygen is released and the recovery of the oxygen ligand happens. Overall, what we are observing here is that kind of step-by-step, very careful oxidation of the metal cluster using the manganese 3 and manganese 4 oxidation state changes, going from S0, S1, S2 to S3 state. Then the very last moment, likely manganese 5 or manganese oxyl-like very active super oxidized species comes, and then go to the O bond formation steps. In a way, this is not really new, but there has been a hypothesis, but we have a way to visualize this kind of chemical processes now. So what it means, I said this step, carefully manganese cluster goes to accumulating the oxidizing equivalent. And that step avoids the creation of these hydroxyl radical type species. So if we put the high energy, we can make a reaction faster. However, there's a risk of creating these highly active radicals. And oxygen evolving complex, or PS2, is what it's exactly doing is to avoid such kind of creating such kind of active species. Then, why did nature choose manganese to split water? So, to be summarized, manganese is the third abundant transition metals, and the number of accessible oxidation state of manganese, it can be manganese 2+, 3+, 4+, and 5+, close to the water oxidation potential. In addition to that, manganese nuclear complexes, like particularly manganese 3 and 4 oxo complexes, is very stable, and it's good for accumulating oxidizing equivalent step by step. And the fourth is that superoxidized species like manganese 5, or it can be manganese 4 oxyl radical type species. This can work as a capacitive discharge process to make a ore bond formation. So I've been talking about oxygen evolving complex. Okay. But what we see also is that how this entire protein is accommodating this kind of very tough multi-electron, multi-proton chemistry. For example, we see the extension of water or proton channels. So these are all the water channels, hydrogen bonded networks, extended from the oxygen evolving complex to the bulk. And one of them, this green chloride, we call chloride 1 channel. We think that this is used for the release of protons. And probably the red one here, oxygen O1 channel, is used for taking substrate water into the oxygen evolving complex. So the point I want to make is that these release of protons and take of substrate into the oxygen evolving complex is well controlled by the hydrogen bonding network, protein environment, spatially as well as temporally. Another fascinating thing, again, it was mentioned by Chris, is that this photosystem 2 has embedded repair mechanisms. Again, I'm not going to the details of this, but I will focus on the assembly of the manganese cluster. So manganese cluster, intact manganese cluster needs to be assembled at the very last step of the PS2 photosystem 2 recovery. And that happens using the manganese 2+, highly soluble manganese 2, under the presence of calcium 2+ ions. And this is not chaperone coded. It can happen with the light. So, which means that assembly of the manganese cluster, manganese calcium cluster, start with the light-driven oxidation of soluble manganese 2+. And the cluster can be self-assembled without any complicated protein machineries. So, coming back to the previous, why did nature choose manganese to split water questions? Manganese 2+ was available in an environment, and it looks like, looking at this manganese chemistry, each manganese oxidation state is taking a certain role. So like a 2+ is important for the assembly process, and manganese 3+, 4+ is important for making a sort of stable multi-nuclear clusters. And then the final ore bond formation reaction, it requires high valent manganese species or oxyl species. So everything is there during the for the manganese chemistries. And some literature, this was think Rutherford and Cardona groups studies, they show that early reaction center likely have an ability to oxidize manganese 2+ species. So manganese is important, but without calcium, this reaction doesn't work. So what is calcium 2+ doing during the reaction and why it's necessary? So the potential role of calcium 2+ is that it is the substrate water it binds 2 water molecules, and also it acts as a Lewis acid, making water electrophilic, and it looks like taking an important role of photoactivation process. Okay. There are other cations. Why it has to be calcium 2+? I think first point maybe it's ionic radius. It's not too big, it's not too small. So calcium radius is about 1 angstrom. Actually, strontium also works. Strontium can be substituted with calcium, and manganese strontium cluster works, but the reaction is not optimized, and the reaction is very slow. So which means that this calcium radius seems to be, and its Lewis acidity seems to be beneficial for the water oxidation reaction. And water exchange rate at the calcium may play a role. And the third point is that evolutionary perspective, calcium binding site probably was already there in the ancestral reaction center before manganese 2+ is involved. Looking at the various photosystem 2, there are many changes. For example, extrinsic subunit is different in cyanobacteria, diatoms, and green algae. But the fact that this manganese cluster, 4 manganese, 1 calcium cluster never be changed, showing that this cluster seems to be optimized for the multi-electron, multi-proton water oxidation reaction. What can we do if manganese is not available? So that's related to the artificial photosynthetic system. Manganese calcium cluster is a nature's choice, which is optimized for pH 6 mild acidic conditions. When we do artificial photosynthetic systems, under acidic condition, what works best is the ruthenium and iridium oxide. However, iridium is one of the rare elements, and ruthenium, this is unstable under oxidative conditions, so it's not perfect. Under acidic conditions, there is an iron nickel mixed hydroxide complexes which can work, but again, this requires high over potentials. So what is my conclusion? So photosystem 2 and the oxygen evolving complex is a nature's blueprint of a sustainable oxidative catalyst. And I would say, as of now, it's perfected over 4 billion years, powered by sunlight, built from abundant element, and still without equal catalyst we are finding chemically. So with that, I want to thank all my colleagues and the collaborators, and thank you so much for listening.
Great, thank you, Junko. Questions?
Hi, thanks for the great talk. I was wondering, before the calcium-magnesium cluster evolved to be able to do all those intricate steps that you outlined to be able to split water, do we know what its biochemical function was?
You mean from of manganese or the, yeah.
Yeah.
So manganese chemistry has been used for like a manganese SOD, superoxide dismutase, or catalase. both of them involved in how to deal with oxygen chemistry. One work for superoxide, and the other one for peroxide. So manganese is already used for the chemistry of oxygen-related reactions. Is that okay? Thanks.
Questions?
Yeah, hello, great talk. My name is Bo. I wondered, you mentioned other atoms that you could potentially replace manganese with. What about the calcium? Is there also an option here for lanthanides? I know some enzymes that use calcium that can use lanthanides. Maybe there's something here as well.
I think it only works with the strontium. So calcium can be replaced with the strontium, but the reaction is slowed down. The activity gets lower as well.
Okay, if there are no more questions, let's thank Junko one more time.
And we will come back at what time? Do you have that in front of you, Nancy?
The time. Oh.
So we'll have a whole 20-minute break, so not to rush this time. Starting at 4 p.m. We should with a very productive seat.
Yep. So come back at 4. Thank you.
This thing doesn't seem to have been on. This thing doesn't seem to have been on.
Mm-hmm.
Is there an on-off switch on this thing? I went to the back of the room and it's not. Is there an on-off switch? Okay.
I don't know. I haven't used that yet.
Yeah, sure. Oh, yeah, you sure? Maybe
Because that would
want to do
be
it.
the second one.
Sure. You want to do it break?
Right now, I
Or
don't know,
okay, maybe after dinner.
After dinner.
Okay.
That's fine.
This thing's not on. Oh, it was not on. Testing. Okay. Testing. Okay, it wasn't on.
Really? It just wasn't on the whole time.
It wasn't on. That's why she was so quiet.
Why didn't
Nobody else
anyone say anything
because they could hear her in the back. No, they can hear her through here. But the room speaker is on. Okay.
And I'm surprised no one said anything.
Yeah, no, because she seemed like this quiet talker. Because I'm sitting here, I couldn't tell. But when I went to the back of the room, then I could tell. It's just,
Yeah.
Okay.
Oops.
Okay.
How do I share just this?
So you go to the Chrome and share, and then go to Window. Actually, sorry, it's so dim.
This
Window.
Yeah, it's in Window.
It's in Window. Okay,
Yep.
then pick that slide. Yeah. Oh, is it in? Wait, it's not in slideshow mode. Put it in slideshow mode first. Go back to the PowerPoint. Put in slideshow mode. Brightness. There. Okay.
Oh, did I share? Or
Okay.
yeah.
Why is that guy always the one showing?
And then
How do we get this guy not to show?
is it because his mic? Well, anyway.
Okay, share. Go to Window, and then there. Oh, it's kind of slow. Wow. Okay. All right.
It's funny. As soon as it's not your computer, you
Yeah.
just lose it.
Okay. Let me do this.
can't make us, you couldn't put this cluster that's working in China.
But it obviously would come from us.
Well, the stories are almost what's left. What's questions are still unanswered. What are there? Are those just questions of governance and this is mechanism? You see the damping of the oscillations. That's always been explained. It's always more than just and this is is if you could overcome that with the oscillator, or is there there's an intrinsic loss of perfect conditions.
There's an old experiment of Cook that if you give one flash and get everything up to both S1 and S2, and then let it sit for a while, it'll come back to S1. And then if you start your train of flashes, you'll get a super oscillation. You'll get a higher oscillation than you would if you started with completely dark method.
And does the stability of the cycle fit the probability of it reminds me of what you would see how frequently chlorophyll is that number because the cycle runs pretty slow. You're able to 1200 to store all that charge, but it'll actually you can store it longer. That's the fastest it'll go. Faster the cycle goes. The fastest you can turn it all the way. You have no limitations to life. 1.2 seconds. But it would never be fast.
All the other
necessary to come with it. So you look where it is. What if you wire it? The central tension versus the account. You don't have. And so, so it is all on one side. Yeah. It's the when I opened the structural work in my body, I said the central tension so that they had a square complex. And so I don't have to yeah, that's what they say they found, but they also have. What's that look like? These things have shown that electrons from got 2.4 electrons. You couldn't do it with less than 3. And then one of them, what we plugged is very interesting. I always needed this was a whole behind like simple weird items. And it was like one of the is for plug your eyes away from the I can probably not go for it's like, all right.
you can see it's really difficult to of those antennas.
I know structural models work because I'm trying to make sure I don't
Sorry. Oh, wait. So let me give it to
instead of light photos. If you had set up something ancestral that's super flexible.
So we just download all slides onto this laptop, and then we share from here.
Oh, okay. So,
is that okay?
I don't need my.
You don't need your laptop.
Okay.
Okay. If you want to use yours, it's possible, but for the sound has to go here and has to go to the people online too. So we're doing.
Okay.
Oh, can I say a question really quick? I had a very minor error in my flash talk slide just with the year of one of my citations.
Oh, okay.
Do you know if I would be able to change it really fast?
Sure. Let's just open it right now.
I'm so sorry
No,
about
let's
that.
open it right now. Okay, it's really annoying.
If not, I'm sure that it's
Oh,
okay.
no,
but-
here, we got it here. Flash talk. There you are.
Thank you so much. It just needs to be 19 instead of 21.
Oh, okay. Not a big deal. Okay. There we go. Great.
Thank you so much.
Sure. Okay.
Small but significant.
Significant, different, yeah. Okay.
It's evolving toxic byproducts to its neighbors. And then later on, it figured, hey, I could use my killing machine to but an electron, so speculation, I would say, that's something that evolved to be able to hold on all times. Idea that flexibility and I think it probably is the way. Yeah. Doesn't become Daniel's water oxidation even if it does make it die. That's faster than Oh, yeah. I think there's three interesting things about one, I have a general argument that a lot of things that are singular are pieces in a way that's that there's a general argument that a lot of them are that way, even if you can't. So I think it's really
Right, then
the oldest splits in between.
I mean, that's, it's like, in this case, I'm like, oh.
That's you have to make that that's again, I think that is specializing in good one here and number three.
I think the oldest splits are surely
Yes.
I think that once you're oxygen, once you're oxygen, once you're switching oxygen.
Oh, this, can I plug this in? Does this have another port, or is that it? That's the last
Are you
port.
sure you want this? It's not bright. People can't see it, so I've been using mine here.
Oh, this is for the clicker, for advancing
Okay.
the slides. Will this advance slides?
Yeah. It's the same thing.
Oh, okay, great.
If you want, I mean.
Do you have another port where you can plug it in?
Oh crap.
Maybe not. Okay, they'll just use they do arrows then. Oh wow, that's so much better. Oh, that's better. Oh my
Yeah.
god. Okay. All right, let's put this out of the way.
So I've just been leaving it up here.
Okay, thanks. I didn't realize that was I've been handing them that other one.
I just don't think molecular cost more than
Absolutely. I think all the stuff for millions of years is all that fantasy.
I think all molecular cost is older than a million years. But you always push out one or another.
C14 brings up
and also like if you look at just all the stuff coming out.
Everyone, we're gonna get started in about 5 minutes.
There's a general thing that that evolutionary are always explosive at the beginning and slow down. When a new selective pressure appears. So I just think it can be that old molecular process, many major innovations with changes. Basically, this place.
But just like this, it's clear it has to be very rapid evolution and then it sticks. Because you look at, like Chris said, look at the cyanobacteria to a sequoia. No, it's not. I think it's the they're locked in. They rapid evolution optimized, and then they just said, nope, we're
I
done.
mean, one of the three most forces in macromolecular complexes on Earth. So no, I would say it's ribosomes, proteases, and
Well, Rubisco is an example of a crappy enzyme. Okay. It's not very, no, it's a crappy enzyme in the world that oxygenates yes. It was a great enzyme before.
Wait, I think three. I third reason that I think that might not be so hard, but I think oxygen is easier than it looks, and that is. And it's this is more about a difference between the way that evolution within oxygen proteins and antioxidant you change their ecology by plugging the reaction centers into other modules.
Is the clicker not working? Someone said the clicker wasn't working.
It's because one's not plugged in, so that would explain it.
So we're good?
I wonder who's. Hey, Nancy. Nancy, were you saying the clicker wasn't working to someone? Were you saying the clicker wasn't working?
Oh, this clicker? I couldn't get it to advance the thing, but we're gonna forget about it. We're just gonna let them use the arrows.
We don't have a.
Alright, so let's turn this on for you. Put that
How do I go back to?
So you're either in the slideshow, you're either in Chrome, or you're in the So here's PowerPoint. So you go into slideshow mode.
Yeah, I did that.
And then sometimes it's a little slow. And then you go into Chrome.
Yeah, that's what I had.
Okay, let's do this again. Chrome. Where's the Chrome? It disappeared. Oh, here's down here. Okay. All right.
Oh, okay.
So okay, go into slideshow mode. Let you do it.
Yeah,
Slideshow mode.
No, I had just
Okay,
lost
yeah,
Chrome.
Yeah, it got hidden.
Are you ready?
Okay, we're ready to start in 1 minute, so.
I'll give you like a 5-minute. Please make your way back and we will get started.
Testing.
Okay, and we will start with our third keynote of this section, Usha Lingappa.
Hi, I'm Usha. I am currently a postdoc with Sabia Merchant at UC Berkeley. But the work that I'm going to talk about today is stuff that I worked on during my PhD, which was with Woody Fisher at Caltech. And it concerns the origins of oxygenic photosynthesis. So oxygenic photosynthesis
Go into PowerPoint slide. Oops. There, now you're.
Oxygenic photosynthesis was a truly spectacular feat of evolution, not just because of the way it transformed everything for life on Earth, but also because it was not easy to evolve. It requires some pretty extreme chemistry, and the leap from other versions of phototrophy to oxygenic photosynthesis is big. In particular, there are two aspects of that leap that I want to highlight. The first is that water has a very high reduction potential, much higher than the electron donors used for anoxygenic phototrophy. That means it is much harder to take electrons off of. So if you want to use it as your electron donor for photosynthesis, you need a reaction center that can generate reduction potentials that are even higher, right? That can access those electrons from water. In photosynthesis as we know it today, this is of course photosystem 2. The second problem is that oxidizing water to dioxygen is a 4-electron process, while photochemical charge separation in a reaction center is inherently a single electron process. So if you want to use water as your electron donor for photosynthesis, in addition to needing a reaction center that can access ridiculously high potentials, you also need some way to bridge it to the 4-electron chemistry of water oxidation. And the way that photosystem 2 does this is with the manganese cluster, the cluster of 4 manganese atoms right at the active site of photosystem 2, which Chris and Junko both already introduced today. Basically, what this cluster does is cycle through oxidation states to store up 4 electrons worth of oxidizing power over the course of the catalytic cycle, serving as that 4 electron bridge. So these are 2 key features of photosystem 2 that set it apart from all of the other anoxygenic reaction centers and are really what enables it to oxidize water. So these are the crux of the problem of how did oxygenic photosynthesis evolve. Then there's this whole flip side to the problem, which is not so much how did it evolve as much as how did life survive evolving it. And that is because oxygen is toxic. It is so reactive and such a strong oxidant, and it engenders other reactive oxygen species like superoxide and peroxides, which are even worse. These molecules wreak havoc for biological systems by causing oxidative stress. And of course, today, we have evolved some pretty sophisticated mechanisms for combating oxidative stress, which allow us to thrive in our oxygenated world and take advantage of the energy available in breathing oxygen. But at the time when oxygenic photosynthesis first evolved, most of those protections did not exist yet. And because of that, the introduction of oxygen to the environment probably caused a devastating mass extinction. But somehow, the ancestors of the cyanobacteria, the ones who evolved this process, right, who would have been the very first organisms to encounter meaningful amounts of oxygen because they were the ones making it, somehow they survived. So if we want to understand the origin of oxygenic photosynthesis, we have these problems from both sides, right? First, how do you evolve the really spectacularly difficult process of oxidizing water? And how do you survive making the first oxygen? The hypothesis that I worked on is the idea that a version of anoxygenic phototrophy using manganese as the electron donor was this key evolutionary stepping stone that opened the door to oxygenic photosynthesis. Now, the first big argument in support of this hypothesis comes from how the manganese cluster of photosystem 2 is made today. This occurs through a process called photoassembly, which is a very unusual way to make a cofactor, where basically each of those manganese atoms comes in one by one and gets oxidized by photosystem 2 to assemble the cluster in place. And the electrons that come out of that oxidation go right into the photosynthesis electron transport chain. They're indistinguishable from electrons that come out of oxidizing water later. So at least in the case of those 4 manganese atoms, we already know it's possible, right? We know manganese can serve as a direct electron donor to photosystem 2. And we know this had to predate oxygenic photosynthesis, right? Because you need to be able to do this first. You need to be able to make this cluster before you're going to be able to oxidize water. Another compelling argument for this hypothesis is the fact that it allows you to separate these first 2 hurdles and solve them one at a time. And this is because manganese redox couples also tend to be very high potential, but are more amenable to single electron oxidations. So we can envision a scenario where you evolve higher potential phototrophy in order to oxidize manganese. You're oxidizing manganese 1 electron at a time. At some point, you stabilize the cluster. That gets you your 4 electron bridge that ultimately empowers you to start oxidizing water. Okay, so that's the hypothesis. To test this hypothesis, we took multiple different approaches. Jim Hemp, who was a former postdoc in Woody's lab, led an effort using comparative biology of photosystem 2 with other modern reaction centers to develop a line of genomic and structural biology arguments. I'm not going to go into this stuff today, but I wanted to mention it just so you know it exists, and I'm happy to talk about it more later if anybody wants to ask. What I am going to talk about is the other things we did. So we looked in the geological record for ancient evidence of this biochemistry. And we did experiments with modern cyanobacteria to kind of empirically ask what vestige of this process they might retain from their ancestors. So starting on the geological side, what we did was we traced back the history of manganese in sedimentary rocks of the Transvaal supergroup in South Africa, which records a phenomenal record from the late Archean through the Paleoproterozoic. So what's going on with manganese that we might be able to see in these rocks? So manganese is abundant,
It's the third most abundant transition metal in the Earth's crust, and it's present there in a lot of igneous minerals in its reduced form, which is manganese 2. That manganese can get released by processes of weathering and erosion, and as long as it stays reduced, it is very soluble in water. But if it gets oxidized, it will precipitate out of solution as insoluble oxide minerals. But remember our redox tower, right? Manganese has a pretty high reduction potential. It is not easy to oxidize. And really the only chemical species in the environment that is a strong enough oxidant to oxidize it is dioxygen, or other reactive oxygen species derived from dioxygen. So in that sense, there's some kind of neat symmetry here, right? Like insofar as we think manganese was the stepping stone for the world to know oxygen, oxygen in turn totally repurposed how the world knew manganese, right? The manganese biogeochemistry today in our oxygenated world is all about redox cycling, but all of the oxidative processes here touch oxygen in one way or another. So in the absence of oxygen, all of this dissolved manganese just stayed reduced, and it accumulated in natural waters as a dissolved species. And concentrations of manganese in the ancient oceans got really very high. We're talking like up to maybe like 100 micromolar. Now this is important and relevant for our story because prior to the advent of oxygenic photosynthesis on the early Earth was electron limited. It wasn't limited by nutrients the way we think about photosynthesis today. Life didn't have the chance to grow to the point where it ran out of any particular nutrient because it always ran out of its electron source first, right? That is part of why oxygenic photosynthesis was so transformative. Unlocking the ability to use the abundant resource of water as your electron source enabled primary productivity to take off in ways that would have been inconceivable before, right? Before, you're relying on a geochemical trickle of electron sources like iron, but iron was pretty depleted from surface waters at this point. So life was really hurting for a better electron source. So this pool of electrons in this reservoir of manganese that's accumulating in the ocean represented a fantastic ecological opportunity, right? There is your incentive that sent life down the path of evolving a higher potential reaction center. Okay, so to simplify this picture into just what we see in the geological record, in the absence of oxygen, all we really get is trace incorporation of manganese 2 and calcium carbonate minerals, because manganese can substitute for calcium in like marine calcite cements, and with an equilibrium partition coefficient, that can give us a proxy for its concentration in ancient seawater. Once there's oxygen in the picture, manganese sedimentation looks totally different, right? Now we have a powerful chemical oxidant that can oxidize all this manganese 2, and it crashes out of solution as manganese 3 and 4 oxides. So suddenly we have much more deposition of manganese in sediments. The biggest example of this type of manganese deposit is the Kalahari manganese field, which is part of the Transvaal supergroup. And this manganese deposit is truly wild. It contains something like 77% of the entire world's high-grade manganese ore. And it is interpreted as a consequence of the rise of oxygen, right? Of all this manganese that accumulated in the ocean, now able to get oxidized and crashing out of solution. So before I joined Woody's group, Jenna Johnson worked on this project as part of her PhD. And what she did was basically look back in time from this giant manganese deposit and ask, did the deposition of oxidized manganese start in lockstep with the rise of oxygen, or was there earlier oxidation of manganese, which you might expect if there was manganese oxidizing photosynthesis before oxygenic photosynthesis? And what she found was pretty good evidence that there is oxidized manganese depositing before the rise of oxygen and co-occurring with pretty good proxies for the absence of oxygen, like the deposition of detrital pyrite grains. So if we go back to these little models of the manganese cycle, there's kind of a missing link here, right? The oxidative branch of the manganese cycle is kickstarting in the absence of oxygen. And this observation we think is best explained by the manganese photosynthesis hypothesis. So after all this work during my PhD, we followed this up with newer drill cores from the other side of the craton, where we have the same intervals of time, but different depositional environments. So while on the one side, you might have deep water environments where you capture more chemical sedimentation, right? Things like these massive manganese deposits. On the other side, you can have much shallower, like shoreline type of environments where you might be able to capture different aspects of the history. Things like more direct, like physical evidence of life in the photic zone. And that's exactly what we found. So this is a spectacular unit of stromatolites. This is a sample from this rock. Stromatolites are commonly understood as basically like fossilized microbial mats. And this is a synchrotron X-ray map of the manganese in this rock. And you can see it is chock full of manganese. This is like tens of weight % manganese in this rock, and it defines the layers of the stromatolite, but is not in, I know it's hard to see in this resolution, but it is not in like detrital sand grains that filled in around it. Meaning this manganese didn't fall here. It precipitated in place, cementing the layers of this map together. So if we were to imagine like what could be the best possible evidence of what we're talking about here, right? Of ancient photosynthetic microbial communities that were all about manganese. I mean, this is kind of it. So from the geological record, we have chemical evidence of manganese oxidation in the absence of oxygen, and we have physical evidence of these photosynthetic microbial communities that were literally lithified in manganese. Then, to test this hypothesis on the biology side, our logic went something like, okay, we think this is a thing in the ancestors of the cyanobacteria, so maybe their modern descendants still retain some vestige of it. This was kind of a crazy thing to look for. Like Tim said, people have been looking for anoxygenic photosynthesis using manganese oxidation for a very long time. And frankly, this is one of very few microbial metabolisms that like should exist on paper, but has been quite elusive. But one of the places that no one ever really looked was in the cyanobacteria, and really pretty specifically, because when you enrich for anoxygenic phototrophs, standard protocol is to add inhibitors that will exclude cyanobacteria, because if you don't, they will outcompete the anoxygenic organisms that you're looking for. So we looked. Basically what we did was we set up simple experiments where we gave them a spike of reduced manganese and measured the production of oxidized manganese over time. The gray line at the bottom here is a control condition of just abiotic chemistry. This is the same culture media, but with no cells. And you can see abiotic manganese oxidation is not occurring under these conditions or on this timescale. Whereas in the presence of cells, we have a robust manganese oxidation phenomenon. Now, while manganese photosynthesis has not been definitively shown before, biological manganese oxidation certainly has. It is not uncommon. The difference, though, is that the other mechanisms of biological manganese oxidation use oxygen, or actually more often superoxide or peroxides, but in any case, some oxygen species for that manganese oxidation. So we wanted to try to rule out these other mechanisms of biologically catalyzed manganese oxidation. So we added the oxidative stress enzymes, superoxide dismutase and catalase. These enzymes will quench the reactive oxygen species, superoxide and hydrogen peroxide, respectively. So if the oxidation we're seeing is only happening via those reactive oxygen species, adding these enzymes should diminish the effect. And that's not really what we see. Even in the presence of high concentrations of these enzymes, we see robust manganese oxidation. We also did the same experiment in the dark and in the light, but with DCMU added. DCMU is an inhibitor of photosystem 2. And both of those conditions completely eliminated manganese oxidation. So we have a manganese oxidation phenomenon that is light dependent, that is photosystem 2 dependent, and that is not inhibited by SOD or catalase. The next thing we did to try to further pinpoint this mechanism to photosystem 2 was use mutant systems. So the first mutant here, the D1 delta mutant, has all 3 copies of the PSBA gene knocked out, so it has no D1 protein, so no functioning photosystem 2. The second mutant, the D170A, has just a single amino acid change right next to the manganese cluster that prevents it from assembling the cluster. So it cannot do oxygenic photosynthesis, but it otherwise, in theory, still has a functioning photosystem. And when we did this experiment with these mutants, we found that the delta mutant cannot oxidize manganese, while the D170A mutant can. So again, providing further evidence that this mechanism is photosystem 2 dependent and is not dependent on local oxygen production. The other approach we took was a more reductionist approach, where we prepared purified thylakoid membranes as like a cell-free system for studying photosystem 2 activity that is more physiologically faithful than like pure protein, but much simpler than like a whole cell. And with the membrane system, we saw a very similar phenomenon as we did with the whole cells. And our collaborator, Hope Johnson, took this a little bit further and did the same experiments with purified photosystem 2 protein and saw a very similar effect there too. So we have data on 3 levels of biochemical complexity, plus these mutant studies and inhibitor studies that together, I think, provide pretty compelling evidence that cyanobacteria oxidize manganese using photosystem 2. Now, along the course of this work, I was broadly interested in the relationship between cyanobacteria and manganese. And I came across a paper that came out of Himadri Pakrasi's lab like 20 years earlier that reported that cyanobacteria, Seneca cystis specifically, the organism we were doing all these experiments in, that Seneca cystis accumulates a huge intracellular pool of manganese. And I was very intrigued by this, so I followed it up. I repeated that experiment, and not just with Seneca cystis, but with the other cyanobacteria that I happened to be working with at the time, which included Gliobacter, which is like the deepest branching member of the photosynthetic cyanobacteria, and a couple strains of Crococcidiopsis, the extremophile that Barry mentioned, and found that all of these cyanobacteria accumulate a huge amount of manganese in their biomass. This is ICP-MS data presented as a ratio to sulfur, just to normalize to total biomass, because some of these strains are very clumpy and difficult to get accurate cell counts on. But for Seneca cystis, this is equivalent to about 10 to the 8 manganese atoms per cell, which is exactly the same ballpark as that original paper, and is just a bananas amount of manganese. If you average that over the cell volume, that's like 100 millimolar manganese. For comparison, I have E. coli and Shewanella up here, which are just as an example of like more normal bacteria, where manganese is much more of a truly trace element. So to dig into what this huge pool of manganese was about, we investigated its speciation using paramagnetic resonance techniques in collaboration with Brian Hoffman's group. And what we found was about 95% of this manganese pool is not protein bound and is instead in these high symmetry, low molecular weight complexes. So these are EPR spectra. This like 6-line hyperfine pattern riding on these relatively narrow wings is indicative of this kind of speciation, as opposed to like low symmetry complex with highly chelating ligands like EDTA or like protein bound manganese, where you're going to get a lot more material in these wings. Here's our cyanobacteria. I only have Seneca cystis here, but all of them, their spectra looked very similar. Again, I have E. coli for comparison, where you can see a lot more of the manganese is protein bound. This speciation is really cool because it implies a functional physiological purpose for this manganese accumulation. And that is because manganese is capable of some truly extraordinary antioxidant chemistry. So some manganese small molecules, for example, will react with superoxide and cycle to manganese 3 and back again in this bimolecular reaction that disproportionates the superoxide but regenerates the manganese 2. So this is catalytic, not stoichiometric superoxide removal. This is the exact same chemistry that superoxide dismutase catalyzes, but just with small molecules, no enzyme needed. And this chemistry is known to be exploited by life as a non-enzymatic antioxidant system. And in fact, accumulation of high symmetry, low molecular weight manganese is a hallmark of oxidative stress resistance. Most famously in the hyperradiation resistant bacteria Deinococcus radiodurans. Deinococcus hyperaccumulates manganese to a very similar extent as our cyanobacteria and maintains it in very similar speciation. So recognizing this chemistry in cyanobacteria brings us a solution to the oxidative stress part of the origin of photosynthesis problem to our manganese hypothesis. Because if oxygenic photosynthesis evolved in organisms that were already really about manganese because they needed it as their electron donor, that association with manganese might have been the only thing that saved them from getting absolutely fried by oxidative stress when they started producing the first oxygen. Like they would have had a fortuitous, like built-in non-enzymatic antioxidant system. So really what I'm arguing here is that I think manganese was the key to the origin of oxygenic photosynthesis on Earth for a whole bunch of reasons. First, because it characterized the environmental context that incentivized the evolutionary trajectory that led to the development of higher potential phototrophy. Then it served as the electron donor for an ancestral version of this high potential reaction center as sort of like the key evolutionary stepping stone that opened the door to oxygenic photosynthesis. And then it provided this like fortuitous early antioxidant system that saved those early oxygenic organisms from killing themselves by producing the first oxygen and the new levels of oxidative stress they would have encountered at that point. Like this might be the only reason this was not a total evolutionary dead end. And both the manganese oxidation part of this and the antioxidant part of this are reflected in the physiology of modern cyanobacteria to this day. And we only came to understand that by studying them to try to better understand this about their ancestors. So with that, I'd like to thank everybody who contributed to the research I showed, especially my PhD advisor, Woody Fisher, and Joan Valentine, who's my mentor in all things chemistry. Funding sources, and happy to take any questions.
Thanks, Susha.
Impeccable timing,
Great talk, thank you. I guess my question is, with like banded iron formations, you see them worldwide, right? And you only see the manganese deposits like this in the one craton. Where are they deposited elsewhere?
They are deposited elsewhere. I only worked on the one craton, but as I understand it, there is evidence of manganese oxidation in other places too, predating the rise of oxygen.
Thanks. You mentioned that those Deinococcus have a bunch of the manganese, and that helps with their radiation resistance. Are cyanobacteria also like particularly radiation resistant compared to other bacteria?
Some cyanobacteria are more radiation resistant than others. And actually, if you look at so like the extremophiles here have more manganese in them than the mesophile. So it does seem to like the more you have of this, the better off you are. I don't know for like the non-extremophilic ones, how they compare to other bacteria in terms of radiation resistance, but yeah, it's an interesting question.
You said the concentration is kind of similar to what the oceans were like before oxygenation?
Oh, no. That was like micromolar. This is like millimolar. But yeah, very different. Yeah. I mean, that was still way higher than the oceans today, but this is higher than that.
Taro, wherever you are, please go get ready.
Okay, Barry Bruce, I am so excited to see that you brought the Deinococcus story because I've been asking many people for many years, 20 years, about that as I don't know if I would call it a corollary with the OEC, but now you've tied this together in some ways. But I noticed in your EPR there seems to be a slight shift in the field of the signature that you have between Deinococcus and your in your 6803. You know that morphologically, if you look at the Crocophales fossils, I think it's almost impossible to tell those apart from Deinococcus. They look anatomically almost identical.
They really do, yeah.
They also have this ability to undergo binary fission in 3 planes. And many of the Crocosteopsis have exactly the same radiation tolerance. And so here you have 2 things. One's a non-phototrope, one is a phototrope. They look like each other. They lived in the same place at the same time. And they both have high manganese content. What's the connection with the origin of OEC? And nobody can ever answer me, but you're the first person who even took a stab at it.
Yeah, no comment.
Is your time? Yeah. Okay, Kevin Redding, ASU. So great, Rashad. I love the story. But if your hypothesis is right about the use of the low molecular weight manganese 2, wouldn't you have had to have internal membranes like thylakoids pre-existing? Because otherwise there's no reason to import manganese, right?
Gliebacter has it.
Well, no. Because otherwise you would just be oxidizing manganese in the periplasm, right? That's because that's where the acceptor site, the donor site would be. So there would be no reason to bring manganese in if you're using it as an electron donor.
So,
unless you had internal membrane.
To be a little more clear, we're pretty sure this big pool is periplasmic, not cytoplasmic.
That's an important point. Okay.
Thanks. So I'll keep it short. Is it me? I think it is. Yeah. I'll keep it short. We can talk more later. So I don't have a dog in this fight in particular because I still think the burial story I can concede what you're saying and still make it difficult to bury manganese oxides. But just a couple of things to understand. Do you really think there's no evidence for oxygen in the surface ocean at this time and in times before this?
I don't know. Yeah, I don't know if I have a dog in that fight myself. Yeah, I feel a little bit. Yeah, I don't know if I have a dog in the fight of exactly when I think this was happening relative to the GOE. I do. Yeah.
It's a massive body of literature with selenium and nitrogen isotopes, so many things, especially in the late Archean. We can talk more about that, but also you just for the record, you think the Kalahari, the Hadazelle, all of that massive amount of manganese, and to get to the other question, there actually aren't others in India, but not in Australia.
Not huge ones like that, but like what you're presenting on, right? The evidence of manganese oxides from the molybdenum stuff and the cerium stuff and whatever else, like that is evidence of oxidized manganese depositing before there is oxygen in the atmosphere, correct?
No, it's not about the atmosphere. The oxygen is accumulating in the surface ocean. That's where it's all happening. It doesn't have to be in the atmosphere. It's not. There's mass-independent sulfur fractionation. The oxygen is produced in the surface ocean and it first accumulated there, and it's leaking out into the atmosphere, but it's consumed. So just final point, so we can talk more.
Thank you. There'll be a breakout where we can discuss.
Yeah. I mean, these are massive amounts of manganese, and I don't think that's being done by this photosynthetic pathway.
Not good. Okay, we're going to do
okay.
We're gonna so let's thank the speaker again.
Okay. Okay, so hello, I'm Taro Matsuo from University of Osaka. Well, thank you very much for giving me this opportunity to introduce our latest study with interdisciplinary communities. So today I'd like to talk about the coevolution between oxygenic phototropes and the surface environment, especially in the Archean era, and focusing on how this relationship can be expanded to the field of astrobiology. The starting point of my talk is an image taken by Voyager 1 in 1990, and this image, of course, inspired Dr. Carl Sagan, and he left the pale blue dot concept. So, and this concept tells us about the blue-colored ocean is symbolized as a cradle of life in the universe. However, I raised a question about so how
has the Earth been always blue? So through the history of the Earth. The reason why I raise such a question is, so surface environment has co-evolved with life, and especially the oxygenic phototrophs had a significant role of oxidizing surface environment. In parallel, so the major evolutionary innovation through the change of the surface environment could promote the could expand the size of the ecosystem up to the planet scale, and which could be detectable by the HWO and life. And one of the great example is a great oxidation event called GOE. So Tim explained. So, and this event has two significant meanings. So one, from the perspective of astrobiology, so global scale environmental change could be remotely detectable, even from a point source, separating from the geological signal. And another perspective, of course, the evolution of life. So the GOE could promote the birth of aerobic respiration, such as eukaryote and multicellular animals. However, we have not well known so how cyanobacteria induced the great oxidation event, also how cyanobacteria evolved in this era, so around GOE. So one of the interesting features for cyanobacteria is their light harvesting antenna called phycobilison. So phycobilison employ the special pigment called phycobilin, so to absorb the green and orange light for photosynthesis that fully complementary with the chlorophyll A. So, but however, so in order to transfer the energy from the phycobilison to the reaction center, so phycobilison require at least 2 or 3 pigments, and leading to the more massive and sophisticated antenna. So now we know the big question to this. So why did cyanobacteria apply the phycobilin pigment rather than the universal pigment chlorophyll A? So the key for understanding to this question is their light harvesting light environment in the Archean era and also modern era. So of course in the modern era, the oxygenic phototrophs have fully developed their own antenna to adapt to their light environment. So and in the Archean era, there might be a different light environment from the modern era. So in fact, so there could exist various minerals so formed through the redox reactions in oceans and atmosphere. So during the gradual oxidation in the oceans and atmosphere, oxidation of the surface environment. So one of the good example is the blue tide in Tokyo Bay, so which is also induced by the redox reactions in the oceans. So let's look back to the Archean era. So the surface environment were fully reduced, including the water environment. And so the reduced elements, such as Fe2+, could be fully dissolved and spread over the entire ocean. So, but once the cyanobacteria emerged in the surface water, so the surface water has been gradually oxidized, and then so reduced iron can be oxidized and turned into the iron hydroxide, and finally leading to the formation of binding BIF. So since the iron hydroxide efficiently absorbs the UV and blue light, so only the green light remained under the oceans. So we have tested this scenario so through the field experiment around Io Island, where the hydrothermal vents supply the Fe2+ and also theta. And so actually the Fe2 plus is rapidly oxidized and then so turned into the iron hydroxide like this. And we have conducted the measurement of so the transmission spectrum, measurement of transmission spectrum under the ocean. And fortunately, we confirmed that the green light environment forms under the depth of the 5 meter here, and which also were reproduced by our model. And after, so I do not go to the biological test, but after performing the phylogenic analysis and also biological experiments simulating Darwinian evolution, so we got one coherent scenario connecting the underwater transmission spectrum and the evolution of the light harvesting antenna. So after the emergence of the first oxygenic phototrophs, the green light environment could form and leading to the formation of Picot zone to absorb the green light. But once the surface environment were fully oxidized, so around NOE, so now the white light environment could spread over the entire surface ocean, so leading to the various antennas. So that is our theory. And then now we apply this scenario to the field of astrobiology. So first of all, we focus on the iron hydroxide. We focus on the one fact that iron oxide could efficiently scatter the incident light to the oceans. And so especially green light can be reflected from the ocean. So it means so the color of the oceans could be constrained to green, but and which could be serving as an indicator for that. But my question is, could iron hydroxide accumulate in surface water over a long time scale? So in order to tackle with this problem, so we have developed the iron cycle model, so including formation, growth, precipitation, and reduction, so in the Archean Ocean. And in addition to that, so we focus on the role of the silica in the Archean Ocean. So we have conducted the growth experiment of iron hydroxide with and without silica. So so we found that the silica, the pure iron hydroxide can be quickly grown up to a few microns, one day after the Fe2 oxidation. So in fact, so as shown in the model panel, so the absorbance of pure iron hydroxide can be significantly dropped after the filtration through a 1 micron filter. So, but in contrast, the iron hydroxide under the existence of the dissolved silica, now the absorbance of the iron hydroxide will keep the same absorbance even after the filtration. And we also measured the diameter of iron hydroxide by DLS, and so most of the particles are less than 200 nanometers. So this means that the silica can significantly slow the aggregation and the growth of the iron hydroxide particle. So and as a result, so based on our calculation, the concentration of iron hydroxide can be enhanced. So especially under the dissolved silica. And in addition, so the silica can also enhance the silica itself can also enhance the albedo of the oceans, especially in the shorter wavelengths. So if the silica could become the particulate form. So finally, we test our models with remote sensing data and also field experiments. So first of all, we extracted the data of the green ocean, clear-colored oceans, and we compared the data with our models. And especially, so based on our field experiment, so we already measured the concentration of iron hydroxide and also silica, and so compared into our model. So this orange model shows is well aligned with remote sensing. Okay, thank you. Remote sensing data. So it means the combination of iron hydroxide and also silica can enhance the albedo of oceans. So especially probably in the Archean era. And I also have a brief comment about the abiotic process. So since iron hydroxide efficiently absorbs the UVC, so photochemical iron hydroxide production can be constrained with a very thin surface layer. So the concentration of the iron hydroxide still remains low, even under the dissolved silica. And now we categorize the abiotic oceans into two cases. So one is modern, like dark blue oceans in the absence of the silica. But if in the Archean era, probably the ocean could become a silica-rich, bright blue oceans. So finally, we got one picture, so connecting between the evolution of light harvesting antenna and the field of astrobiology. And so my main message here is the color of the oceans could co-evolve with the life, especially the oceanic phototrophs. Thank you very much.
We have time for a couple questions.
Yeah, thank you so much for that great talk. Have you considered the climatic effect of this on the planet?
Climate.
It's a very substantial increase in albedo.
Yes, climate is. I just considered the climate effect for the abiotic process only. So we also calculated the the flux of the UVC under the existence of haze condition, and then of course under the haze condition, the abiotic process can be suppressed. But we have not considered how the weather condition affects the abiotic process and scenario. So kind of weather conditions or weather model do you affect our abiotic scenarios? Sorry, I just have a question.
Sorry. Sorry, I meant it would have a net cooling impact.
Cooling impact.
If you increase the albedo of the oceans, they're very dark, and then if you made them very bright, that's
I see. Okay, I got point. Okay, yeah. Actually, Josh pointed out this point. Actually, then maybe the evolution of life could be affected. Yes, by that point. Yes, thank you.
One more over here.
Oh, just a super quick comment. So in Agrisel's earlier talk, she did retrievals on this green ocean, and it turned out because now Taro has shown that these iron particles can stay suspended, this is one of the most detectable surface biosignatures that we found, at least in the initial surface biosignature work. So it's tremendously promising for the Agouron Observatory.
Thank you.
Thank the speaker again. Thank you. So we're going to have a flash talk. So could the poster presenters please line up in alphabetical order? Gonna get a 1 minute flash talk. Okay. All right. So alphabetical order.
Is it alphabetical by first or last name?
Last name, by last name. Okay. So you're going to hand off the microphone to the next person, all right? So you can go arrow, use the arrow to go to the next slide. Okay. And
are we lined up?
Are you buyer? Yes, you're
yes.
buyer. Okay. Oops. Come on. Oh, that was forget this part. Okay. All right. There we go.
Let me get it started. Hello, my name is Allie Byer. Hi, my name is Allie Byer, and I am researching how rhodopsin-based light harvesting can be used to expand life detection frameworks and improve the interpretability of habitable worlds observatory observations. So we didn't talk about them yet, but I think we will tomorrow. Rhodopsins are membrane proteins that harvest light using retinol and are found in almost every corner of our planet. Bacteriorhodopsin absorbs light around 570 nanometers and functions as an outward proton pump in archaea to establish a proton gradient for ATP production. I am proposing a protocell model built on cell-free protein synthesis and ancestral rhodopsins that allows for studying non-photosynthetic pigments as biosignatures within varied environmental contexts. My model is also inspired by Berhanu et al. 2019. Thank you.
Hello. So this is the phenomenon of kleptoplasty in Antarctica. We discovered it in 2007, our collaborator, Rebecca Gast. And this is a phenomenon where an heterotroph can acquire phototrophy by eating the most dominant algal species in the Southern Ocean and retaining its plastids as kleptoplasts. This allowed us to study the same plastid with the same pigmentation in two different cellular hosts. And we saw in the heterotroph host, in the kleptoplast, a really low oxygenic state. And on the other hand, in its original algal cell, we saw a really, we saw a great light adaptation under the imbalanced light of Antarctica and UV excitation of chlorophyll.
Hi everyone, my name is Ash. My poster looks like this. And I want to ask you, have you ever wondered if you could go back in time to the Archean and visit it? If the answer is no, I don't blame you. The weather probably sucked. But if you are curious, and you've heard about iron-rich oceans, sulfide-rich oceans, green oceans, purple oceans, and you want to know what that looked like, I, as an oceanographer, would like to give you a picture of that and maybe convince you in the context of oceanography that those conditions were not global and rather very spatially heterogeneous. And this would have played a big role in the detection of global factors such as visible pigments or global fluxes to the atmosphere, as well as to rates of oxygenic and anoxygenic photosynthesis relative to nutrient supply, and global fluxes of oxygen to the atmosphere as well. So if you're interested, please come talk to me. I'll show you what the Archean oceans look like.
Hi everyone, I'm Sally from UC Riverside. My poster briefly discusses how light harvesting systems on Earth can help us think about extraterrestrial photosynthesis. I focus on 3 key systems, as you see, and as you know, everyone discussed this, they are different in their biochemical structure and absorption range. A key point is that extraterrestrial photosynthesis needs more than a favorable planetary environment and the presence of light harvesting systems. If the star is not sunlight, it may emit a lot of harmful radiation, and so the organism also needs a toolkit for photoprotection, molecular repair, and stress tolerance. As experimental and genomic data expand, we can refine model phototrophs by looking for co-occurrence of clusters of genes in photosynthesis and extreme environment tolerance, stressors tolerance, and this would help us move from a list of interesting organisms to a very well biologically informed biosignature catalog of model phototrophs. I actually designed a bioinformatics pipeline, which I am currently refining to screen genomic and protein databases for co-accurating phototrophy. Yeah, this kind of. I very much hope
There'll be a warning to you all.
All right, I shall try to avoid that embarrassment. Hello, everyone. I am Simon Frommler. I'm from the University of Minnesota, and today I want to talk to you about a new model system that we've been developing in our lab to study evolution in conditions from the past or on other worlds using experimental evolution. So our study system we've been talking about a lot is the Archean, which, as you know, really anoxic, very highly ferrous oceans. So what we want to know is, did these conditions provide evolutionary trajectories that were possible then but aren't possible now? One particular one we're interested in is rhodopsin, which is pretty well suited to the green light oceans we've been talking about. And here you can see a diagram of what we're proposing is a rhodopsin-based form of photoferrotrophy. I know to some of you that might sound a little out there, but the metabolic viability of this has actually already been demonstrated in bioengineering experiments. So what we're hoping to do is just demonstrate its evolutionary viability with lab evolution. It's a bit of a complicated project, so there's a lot more nuance than I'm giving here, so please come by my poster. I would love to talk to you about it.
Hi, I'm Yuko Matsu. I will talk now about the excited energy transfer EET model. And before 10 years, I developed a model of the EET by using the C programming language. And now I'm developing the new Python-based model combined with the quantum chemical calculation with the automated workflow. And for example, when we specify the protein data bank ID, the structure of the antenna system is downloaded automatically and picked up the unit of the photosynthetic pigment extracted considering the rotational symmetry, and after that the parallelized quantum chemical calculation is conducted, and then the result is collected, and time development of the EET is calculated. So currently I'm considering just dipole-dipole interaction, but the more sophisticated interactions will be updated. So I will tell you the detail in the poster. So thank you.
Hi, my name is Tolga, and this is my undergraduate research project, and it is about carbon monoxide on early Earth and its possible relevance on origin of life. So we propose that carbon monoxide produced by UV photolysis of carbon dioxide will accumulate on early Earth atmosphere, and this will establish strong redox gradients and a far from equilibrium chemistry. And this might favor the emergence of primitive catalytical structures similar to carbon monoxide dehydrogenase, which is a central enzyme in Wood-Langdell pathway. And to test this possibility, I used a 1D photochemical model and tested various atmospheric scenarios and found that carbon monoxide can accumulate on early Earth's atmosphere under various cases.
Hey everyone, I'm Mia. I am a second-year PhD student at the University of Edinburgh, and I'm currently looking how the vegetation red edge is not an unambiguous biosignature. I'm looking at how some minerals, specifically silicates and sulfides, can look like the vegetation red edge because of their spectral features. If you want to come talk to me about what I'm doing, that would be fantastic. See you at my poster later. We just recently published our work on arXiv, so that's that little QR code on the right, which I'm also happy to provide later. And I'm also the host of the Tartan Tardigrade podcast. So it's brought to you by the UK Centre for Astrobiology, and I interview astrobiologists about their cool work, about their career journey, and also a bit more of their personal side of what it means to be a scientist. So if you want to have a listen, please do, and I'm happy to see you guys later. Thank you so much.
Hi, I'm Gautam. I'm in the Agrisera lab, and we are mostly this project is focused on the case one of the model system for the far-red light accumulation. Mostly you can see that one, they have two things we focus on this project. One is for the iron apoptosis. You can see they can tolerate a higher iron and lower iron with whatever environment they can grow. And the latter, you can see that if you grow the higher iron, they can tolerate almost more than 10 or millimolar of iron. And also that just accumulate iron deposit, intercellular, extracellular, whatever you can see. And the latter thing for this project, we just try to the CRISPRs want to try to observe the structure of this kind of far-rated core containing for the system 1, and how is assembled with ISA protein. And also later on, we try to study structurally how light harvesting is going energy transfer. Thank you.
Hi, my name is Felisa, and I'm a senior research scientist at Berry. I live actually in space at UCSF, but also in LBL. I'll talk about more on how that works. So I've been interested in resurrecting something that I worked on a number of years ago. I'm basically interested in the flexibility of the life we do know. In particular, knowing that nitrogen fixation and photosynthesis, oxygenic photosynthesis, are in competition. But could there be a way they can compete? If we think about nitrogen fixation, it produces more hydrogen than it does fixing nitrogen. So what if you could recycle that nitrogen, or that hydrogen, through anoxygenic photosynthesis in a cyanobacteria, so turning off photosystem 2? We built this light box that you can see up here. There's a cyanobacteria that I pulled out of an environmental isolate. And basically what you're seeing in the other panel is that it will oxidize sulfide, but maybe it can do some other oxidation steps to do anoxygenic photosynthesis. This has implications that we've heard from many people today, not just in astrobiology, but also in terms of early Earth, or in terms of using light from an enemy dwarf star. Thanks so much.
I'm Mike Wong from Carnegie Science, and I'm showing you what typical plant matter looks like to a flight-ready in situ biosignature instrument called a pyrolysis gas chromatograph mass spectrometer. We can show many different types of samples to our PyGCMS, including photosynthetic microbes, non-photosynthetic microbes, microbes that have long gone, or photosynthetic plants that are now fossilized into coal and shale, fungi, and also abiotic things like meteorites. And now the patterns that you see here, each spike is a different organic compound inside of these carbon-rich materials, and there are about 10,000 of them in each of these plots. Now I can't find patterns in 10,000 dimensional space, but we can train a machine learning algorithm to do that. So what we've done is shown that you can train a machine learning algorithm to identify signs of the patterns of life versus non-life in these types of data sets, as well in our latest 2025 paper, signs of photosynthetic life versus non-photosynthetic life. And if you'd like to learn more about how we do this, you can stop by my poster, which is actually Bob Hazen's poster.
Hi everyone, my name is Anastasia Anchelina. I'm with the SETI Institute. Some of the work I'm involved with now is synthesis of hydrothermal vent analogs, also known as chemical gardens, as a way to mimic some of these systems that we see in our oceans, with also the thought that it's one of the theories for original life here on Earth, and we are hypothesizing that these are the systems that we can find in some of the ocean world moons within our solar system, and likely outside as well. And some of the work, this was not an updated slide, but I updated later, but some of the work I'm presenting is some FTIR measurements that I have from the systems that I synthesized, and they are non-equilibrium and energy minimum systems, and such that you have like a salt seed that you use, that you drop into an alkaline solution, and which creates a membrane. And I'm presenting some results that I made with FTIR, where that showed that depending on the addition of different organics to the structures, there is changes in the absorption bands of the actual complexes. That's it.
My name is Tane. I'm from UW Madison. My topic is so very, very different from most people here. I'm a bit confused. I'm here. Anyway, so I think the only contribution I can make is a showcase of what imaging spectroscopy can do. So what I'm using here is a project we did back in 2019. It's called Cheese Hat because we're in Wisconsin. So this is a 10 kilometer by 10 kilometer Cheese Hat northern wood in Wisconsin. We use imaging spectroscopy to map leaf nitrogen. So we're not focusing on pigments, but we have the ability to map nutrient. By linking our mapped nitrogen to tower measured GPP, we can better model variation in GPP and showing what kind of influence different level of nitrogen have for different patches of forest. So our study shows in order to better model GPP variation, you really need to incorporate nitrogen for very patchy, very diverse forest landscape like what we have on the surface of this. Yeah, thank you.
Thank you, everybody, for wonderful talks and for those poster flash talks. Okay, so now, Bob, can I recruit you to come help moderate the discussion? So we're going to decide on topics for breakout sessions. We can have 3 online if you guys want to break out one that's not online and sit out in the patio, you can also do that. But so what we're going to do is I'd like to take suggestions from all of you. And could we have a microphone runner volunteer? Bob, can you come over here? Use this. Actually, I'll use this mic. What? Oh, just feel the questions. So what I'm going to do is Bob is going to take your questions. I'm going to be typing them down. So let's see. Anybody have a particular burning topic they want to discuss with lots of people in a room? Origins of something? Oh, Felisa?
Read the list of like all the elements we're talking
about.
Iron and oxygen.
Okay.
It's like who's on first? It's like whether you're a protein person or you're a pigment person.
Okay.
Or metals person, you know what I mean?
Okay. All the elements. So iron, manganese, proteins, pigments, nitrogen.
Calcium.
Okay.
Elements, your favorite element?
Any other burning topics? Just sulfur. But one breakout topic, or should we break this into different breakouts?
Yeah, just trying to go from first principles to get started.
Okay, any other ideas? Oh yeah, okay, sure, I'll do that. Let's see.
We all have our pet questions for this.
Okay. All right. Any other ideas? No?
I noticed the theme in a lot of the talks where many of us are interested in how the planet and the life on the planet co-evolve. So I think that would be a really good breakout topic.
Okay.
I don't know what you want me to do.
Okay. Because I'm typing, so you can just call out people. Thanks. Okay.
The obvious ones that keep coming up again and again, but like all the bottlenecks to getting to oxygen in the atmosphere, like making anisogenating photosynthesis, oxidizing water, accumulating oxygen in the atmosphere because you bury carbon, et cetera, et cetera. So all the things that have to happen in order to even detect that. And maybe to believe it too, as it's been brought up. Might as well put it on the list.
Okay. How about maybe we break that down? One origin of the OEC, one origins of chlorophylls, maybe pigments.
Yeah, those are good.
Okay. All right.
Good thing.
All right.
It seems like the manganese in the OEC is certainly a major question, and the pigments are separate, somewhat separable question.
Okay. Any other?
I don't think I captured it, but I think comment was like, what were the limits to the OEC? The origin and limits of OEC.
Okay.
And then the one I was interested in is light. Properties of light.
Okay.
Property. Maybe in their own.
Do you want to expand on that a little bit more? Properties
Like
of light.
NIR, flux, star types.
Okay.
Probably detectability as well for that. But the one I was going to mention also kind of like obvious is like the false positives from like the true negatives and stuff.
I'd like to also have a discussion with people who aren't astronomers about how a planetary environment and the stellar environment can potentially affect photosynthesis in detectable ways.
Okay. How planet would that come under coevolution or no?
Coevolution.
Okay.
Get all the way
Okay.
to, what does the planetary environment do to photosynthesis and how would that change what we're looking for?
Shape.
This might also fall under the same one, but I'm interested in, if anybody has strong opinions on the apparent kind of co-occurrence of the snowballs and the big oxidation events, whether people think that that's a coincidence or what the direction of causality is, etc.
So that's that against co-occurrence of snowball states in. Okay, and oxidation events. Okay, are we doing good? We have more than 3 topics. Shall we? Do we want to consolidate some of these? Vote on them. Yeah. Okay. Let's see.
Can I add to the
Yes.
bottlenecks for oxygenic photosynthesis?
Yes. Yeah.
And say the bottlenecks for sustained detectability of oxygen in the atmosphere.
Okay.
And more generally, the bottlenecks to sustaining of ice.
Something that I haven't heard the word stated, but lateral gene transfer and genomic shuffling is clearly going to be a case in many of the cases, like the far red light photosystems. Those have to be moved laterally in some way. The retinal pigment proteins we'll talk about tomorrow probably have to have the same thing. The violins and the phytochromes and the cyanochromes, all of that. All of those things had common origins and had to move sideways, and nobody's mentioned that.
Should I consolidate under one of these other categories? No. Let's see. Okay. Any more? Okay. I'll make this bigger. Sorry, it's too small.
Just a
to
quick
see.
follow-up
Yeah.
on this. I see the risk. There are 2 really different groups in here at least.
Yeah.
Maybe 3 different groups, right?
Yeah.
And there's really a risk that everybody's going to end up with their cohort having the same conversations. So I would
Thank you.
structure these in a way that demand mixing, even if you do that.
Okay.
Creating a dating app. You know what I'm saying, right?
Okay. Let's see. Yes. Yep. Okay.
So I think for the lateral gene transfer, it's not only for pigments, but for other requirements for photosynthesis.
Okay.
Yeah, I was just saying that LGT not only applies to pigments, but to other mechanisms or proteins that are important for photosynthesis. Maybe expanding that a little bit would be good.
Okay. Okay. Shall we do this? Who here considers themselves not a biologist? Okay, so you're not allowed to cluster in one room. Okay, so we're going to consolidate this down into three rooms, or 4. We could do 4 if anybody wants to meet somewhere else. Okay, so for properties of light and radiation, I'll do that under star properties. Okay. And let's see. So these are bottlenecks to get to oxygenic photosynthesis. This is the origin. So these, I think these should be two different rooms. And let's see. All right. So I think this question is definitely just could be only astronomer question, could be about limits. Okay, actually this could go down here. Detectability, bottlenecks for sustaining a biosphere. All right, getting down now to 5. Okay. Would this cluster anywhere? Lateral gene transfer. Maybe that's something to do with coevolution. Actually, it's too much. Any suggestions here?
Are there people who would consider themselves experts at identifying the presence of pigments on other planets? Because maybe those people and the more Earth-based pigment people could all be together in one place. Just trying to think of a way to kind of merge some of these topics so it's not only one group.
Maybe the co-occurrence of snowball states and oxidation events can go with origins at OEC. Okay, that'll put detectability.
A lot of these elements in the coevolution planet atmosphere are in manganese.
Okay. Wait.
Doesn't really make sense to talk about.
Okay. Okay, some of these are a little bit too big for one group here. All right, so we sort of have 3 clusters here. Are they clustered okay, or some clusters need to be separated? Okay, I'm not sure that gene transfer and star properties. Yes.
Well, no. So my comment is after this process,
Mm-hmm.
but before we all break,
Mm-hmm.
I'd really like to talk about what the goals of these breakouts are.
Okay.
Because we don't
Yeah.
just want to. I mean,
Yeah.
I don't just want to.
Yeah.
I love sitting and listening and learning.
Okay.
We've been doing a lot of that.
Right.
And if we're getting into smaller groups,
Mm-hmm.
can we have a more focused sort of what's the goal of the breakouts?
Like for example,
yeah.
should we really be considering this in an astrobiology context, like to talk to headquarters? Like, I mean, I agree, give us very specific instructions.
Okay. All right. Think in terms of what will go into a white paper in terms of research agenda. Yeah.
In the context
Yes.
of
For advising. Getting funding. so advising what are research priorities to support. Okay. Is that enough of a goal?
Ideas about other things that could come out.
Okay. Yeah. So you'll come up with some topics here. All right. So we're gonna have. I'm not sure the gene transfer and pigments. They might be a very different topic from the star and planet environment, I think. Maybe, and these 2 topics could be very different too. All right, so, yeah. Yeah, it's very different. Okay, or how about do this? I'll put the pigments together. I suggest we have a topic on origins of the OEC, and that will also include star planet environment. Okay. And if you want to, it'll be under here. Oops. Okay. All right. So it could be the or prebiotic environment. So everything kind of falls under coevolution of biosphere and planet anyway, right? So okay. So I'll just make this as a broad. Let's see, bottlenecks to the OEC. Yeah.
Yeah, I was gonna say the correlation of biosphere and planet. What I wanted to understand is in different environments, right, different stars, how does photosynthesis change detectable ways?
Okay.
There's a very strong detectability. We're pointing towards what should HW or look for, right?
Right.
So I don't want that to get lost as a detectability.
Okay, so we'll put that. How about under the light aspects, star shape? I mean, it's more than pigments here, but okay, I think we got four groups maybe.
I think nutrients could be in biosphere and planet, not just
Okay.
I guess environment.
Okay, nutrient.
Light is also, yeah. So I think Nancy's already said this, but just to kind of state it another way. So the audience for the white papers are, I think, two potential groups. One is this, we have this ongoing DARES astrobiology decadal strategy activity. I'm involved in that, and probably other people are in the room. So it's identifying research gaps and needed capabilities that you can tell the astrobiology program, hey, we need funding in this area. So Nancy will be presenting on that at AbSciCon. The other audience is HWO, the project office, Vicky's on this community science and instrument team. And you guys, you know, what Nancy's done is amazing here. She's brought these people together. And just as Vicky was saying,
It's not just me, Nikki, the whole SOC did this. Okay. I didn't know everybody. Okay.
And just as Vicky was saying, like, we need to learn about different possibilities for photosynthesis, you know, in these different star planet combinations. And you need to tell us what to look for, because right now we're in the critical stage of designing the mission. And if you have input to give saying, hey, you really need to focus on that, that can be translated in measurement requirements for the instrument development. So that may be a little too highly specific. I admit I'm biased, but that's something to keep in mind for output from this workshop.
Okay, I think so. None of these groups is exclusive of the other. So we're definitely everything's related. But I've broken it down now. Definitely, we want to treat star planet environment and bottlenecks for sustaining that biosphere and detectability, and then getting it false positive and negative. So if we keep that as one breakout, and the origins and limits of the OEC, I think it's a substantial, that should be its own topic. And then the origins of the light harvesting pigments and how they've evolved could be its own group. Is everybody happy with that? Okay, so and feel free to bring up any other topics you want, but we'll sort out. So there's two breakout rooms in the back. For the larger group, we'll probably stay here. I'm not sure how many people are going to go to which room. So yeah. Okay, so first for the first, this will be breakout one, so the larger room. Let's see a show of hands who wants to go into that room. Okay. All right. Breakout one. Okay. And don't all the astronomers go there, okay? All right. Okay. Breakout two. Show of hands. Bob, everyone. Okay. All right. That's a little smaller. Okay, a smaller room. Okay. And then all right. Breakout three. Okay, great. Okay. Yeah.
Loosely, it's maybe 25, 10.
Yeah. Okay, so breakout one will be in this room. I think we have the most hands for that. Breakout two, see. So, yeah. So Nikki, Avi, and let's see, you who's. Okay, who else is helping? Oh, and Alessandro will be just making sure everybody's set up. So, but you guys decide which room you want to go to. Which you want to go to 2, Nikki? You go to 3. So, Avi, you want to stay here? Okay, that's happy. Okay, just talking with the astronomers. What? Okay, I'm going to set up so online people will be setting up rooms. might take a little glitches and to get started. And then after that's over, please also, can you please also select somebody to take notes and do a report? We'll have a report back tomorrow, okay? So select somebody in your group to take notes. Okay. All right. Okay.
Which room?
Oh. Okay. So I'm gonna go to controls, breakout room.
Okay, so just clarification, this is breakout 1. Breakout 2 will be the next room over. And then there's an around the corner one that's harder to find. That Alessandro, you're 3. Follow the guy in the purple sweatshirt. It's around the corner, that's breakout 3.
Set that up. Okay, do recording. Stop.